Excellent. Top of the morning to you. It is Tuesday, March 17th, 2021. Today we hit the part two of our digestive system three-part lecture series. So we will talk about the digestive system, the digestive anatomy, and the digestive histology, which if you've looked at your study guides, which if you haven't, first and foremost, shame on you, you will see that there is a lot of histology. Not quite as much as we saw in the endocrine system, obviously, but definitely more than there was in the cardiovascular system. And so the nice thing is, though, if you know what to look for, it can be pretty easy to tell one organ from the other as we work our way through the alimentary canal. We still have two assignments. That sounds right. Yeah, two assignments left. Your PhysioX exercise 8 and your Labster intestinal glucose transport that are both due on Monday. So make sure you have that. And then Wednesday next week is your lab and lecture exam. So this is your big weekend to study this material. Again, it's quick, straightforward system, one organ system that we're going to be dealing with here. So it is fast. Then you get a whole week off to start working on the respiratory and urinary system, which we're going to do when we come back, so that you don't have too much fun on that entire week off. I do want you to complete pre-labs 22 and pre-labs 25 on the urinary system and respiratory system to make sure that we come to school prepared on the 5th when we come back. All right, questions on any of that? All right, excellent then. With that in our pocket, let's dive back into lecture. And what we had left with is, again, this concept of all the functions in the oral cavity. Oops, wrong. I don't want to do that. I want to do this. Again, we talked about how that food undergoes those iterations, those changes. And the first one is to be converted from food into that substance we call bolus, where we have masticated the food, we have mixed it with the saliva into that nice big goopy mess. And then our next function is going to be to propel that bolus down towards our stomach in a process that we commonly refer to as swallowing, but the appropriate anatomical term for is deglutition. Deglutition is a rather elaborate subconscious reflex. What do I mean by that? What do I mean when I say that it is a subconscious reflex? Part of it is that it is involuntary, absolutely. I mean, you could argue that the initiation of it is partially voluntary, right? You are taking the bolus with your tongue and presenting it to the posterior part of the oral cavity. But many of you have probably experienced, if you've been sucking on a Lifesaver or a Jolly Rancher or a piece of gum or something like that, and it went too far back, it may have accidentally triggered your swallow reflex and you swallowed that piece of candy down, right? So absolutely, it is not something we voluntarily control, but it is something we need to be conscious for. Again, as we mentioned, today is St. Patrick's Day. So again, back in ancient times, you used to go out to bars, drink lots of green beer. And if someone passes out and you need to get them to drive you home, is it wise to pour scalding hot coffee into their mouth to help wake them up? No, because this is a subconscious reflex. If you are not conscious, if that person is passed out, then all you're going to be doing is pouring that scalding hot coffee into their lungs, which may wake them up, but definitely is not a good thing to do. And it definitely doesn't feel good either. Excellent. So again, you have to be awake. You have to be conscious for this to take place. This reflex includes 3 main phases, and not surprisingly, these phases are named after and associated with where the bolus is located during the process. So it starts first, as we mentioned, subconscious reflex, with the buccal phase. Buccal phase is, of course, when we're forming that bolus, masticating the food, mixing it with the saliva, moving it around with our tongue. And then what we do is we present that bolus with the tongue towards our oral pharynx. Remember, as we talked about, this muscular tube that is the pharynx basically has 3 main portions, 3 main regions to it. The nasal pharynx, the oral pharynx, and the laryngopharynx associated with the big openings they are adjacent to. And when we present this bolus towards the posterior part of the oral cavity to the oral pharynx, basically two things are going to occur. We see the beginning of that process here. Notice the first thing that happens is that soft palate, that uvula that we talked about, is going to be pushed up. And when it is pushed up, it closes off the nasopharynx to stop the food from going into the nasal cavity. We want to make sure we're directing it into the esophagus. The other thing that happens as we present it to the oral cavity, the posterior part of the oral cavity, is remember those two arches we talked about, the palatopharyngeal and the palatoglossal arches. The bolus is going to come in contact with it. And when it makes contact with the arches, this is what triggers the swallow reflex. So making contact with that is what triggers that swallow reflex. You can experience this swallow reflex by putting your hand on your throat and swallowing. And when you do that, you should feel your laryngeal prominence, which is the prominent portion of the larynx. The larynx has this big thyroid cartilage with that prominent bump that sticks out. That bump is enhanced, its cartilage is enhanced by androgens, which is one of the reasons why males tend to have a more prominent Adam's apple than females do. But females get an Adam's apple in there as well. It's just not necessarily as prominent. But it is going to make contact with that oral pharynx and cause that trigger reflex, which, as you can feel, causes the larynx to elevate. Now, the reason we want it to elevate is let's look at the starting point of what's going on down here. Remember, we talked about how the larynx is that guard that is going to regulate the flow of where food and water and where air goes. And while you're sitting here calmly in your chair right now, you can see that our friend, the epiglottis, if you remember, we talked about that way back in 430. It is made up of elastic cartilage, the same cartilage that makes your ear. So the same rubbery cartilage that makes up your ear makes up this flask called the epiglottis. And it is present in an up position. Notice that opens the airway. So as we breathe, air can freely flow into the larynx, into the trachea, and down to the lungs. At the same time, we can see that the upper part of the esophagus is closed. Like the pharynx, the esophagus is a muscular tube. Like the pharynx, the esophagus is basically just a conduit. It serves one function and one function only, and that is propulsion. And like many of the organs, not all, but many of the organs in the alimentary canal, we want to regulate flow through it. And the way we regulate flow through it is by having a valve located at the proximal and the distal end of it. Again, if you think back to 430, one of the things we talked about is that with a circular muscle, and remember with our alimentary canal, we have a circular layer of smooth muscle. And what can happen is that smooth muscle layer can thicken, so that not only does it change the diameter of the lumen, but that thickened muscular layer can actually form a valve that is capable of closing and stopping the flow of materials through it. And what would we call a structure like that, which was made up of a thickening or enlarging of that circular layer, forming a valve that regulated flow? Exactly, we would call it a sphincter. So up here, we have what is known as the upper esophageal sphincter. And notice that this upper esophageal is closed. So at our starting point, the epiglottis, and therefore the opening, the glottis is open, and our upper esophageal sphincter is closed, therefore our esophagus is closed. So while you're sitting here calmly, air can come in, your soft palate is down, breath can go right back in. However, when we present the bolus to the posterior part of the oral cavity, it closes the soft palate so that that is no longer able to be a pathway for air or for food to go into, and it causes the larynx to open. And that occurs in what is known as the pharyngeal phase. Notice when the bolus reaches that pharyngeal phase, and again, we can see how that uvula of the soft palate has closed off the narrow pharynx, and our larynx, as it elevated, as it raised in our throat, we can see two things that have happened. The first thing that has happened is that elastic epiglottis has closed over the top of the larynx, closed the glottis, which is the opening there, so that the food cannot enter into our airway. And notice also the elevating of the larynx has opened the upper esophageal sphincter. So again, we make contact with those pharyngeal arches. This is a subconscious reflex that goes through the medulla oblongata. Our larynx elevates, our epiglottis closes, and our upper esophageal sphincter opens. At this point, notice there is only one place that our food, our bolus, can go. It cannot go up into the nasal pharynx because that is closed. It cannot go into the airway because that is closed. So the only place it can go is into the esophagus. And so muscular contractions of the pharynx are going to move that bolus into the esophagus. All right. Notice this is all working fine and dandy until you do something like trying to talk, or you laugh, or you sneeze while you're trying to eat. When that occurs, obviously, that forces air out of the esophagus. As it forces it out, probably not out of the esophagus, out of the trachea, opening up the epiglottis. That can force the bolus up into the nasal cavity, right? You get the milk going up your nose from laughing, right? Or if you try to talk, opening that airway can allow the food to enter into that airway, causing you to cough as a result of it. All right, this is why it's important not to talk, not to laugh while you are undergoing this swallow reflex. Now, once the bolus enters into the esophagus, and again, you can feel this. When you swallow, the larynx elevates and then it descends again. And so when it descends is when our bolus has now entered into the esophagus. Notice once it has entered into the esophagus, our larynx is going to descend. And when it descends, those two events that just occurred are going to unoccur. Our epiglottis will open back up and our airway is now open. Our upper esophageal sphincter will now close, stopping the food from coming back up. And our soft palate descends, opening up the airway again. This point, the food is in the esophagus. And does it just stay locked there in the esophagus?
No, it travels down to the stomach.
Yeah, it's going to travel, exactly. And how is it going to travel? By gravity, carrying it downward?
Peristalsis.
Peristalsis. And depending on what it is, take anywhere from about 1 to 8 seconds, right? If it is liquid, pretty much 1 to 2 seconds for it to go down. If it's more solid food, take closer to 7 to 8 seconds. But definitely in less than 10 seconds, whatever it is that you've swallowed has reached your stomach via peristalsis. So again, we contract those pharyngeal muscles. The bolus enters into the esophagus. Whoops. Our upper esophageal sphincter closes. Our epiglottis elevates, right? All because of the, should say this here, our larynx descends. And so when the larynx descends, the upper esophageal sphincter closes, the airway opens back up again. And peristalsis is going to move the food towards the stomach. Where notice there is yet again a second, we'll answer that in just a second, a second valve And this valve is the lower esophageal sphincter. It is also known as the gastroesophageal, because gastro refers to stomach. And the top part of the stomach is also known as the cardial region. So it is also known as the cardial esophageal sphincter. So all three of those are appropriate terms for this lower valve, the distal valve of the esophagus. Now, great question. What happens when someone vomits is basically it can be acidity. It can be certain substrates that are present in the stomach. It can be emotional or some type of top-down cognitive reasoning that is taking place can cause a reverse peristalsis. And with that reverse peristalsis, that exactly causes the food to elevate from the stomach and be ejected in that way as well. No, the esophagus doesn't close in the same way during vomiting, which is one of the reasons why you have to be really careful when someone is passed out and vomits, is that they can actually aspirate some of that, which is why you want to turn their mouth to the side so that they're able to move it out there. Typically, the vomiting process involves not just contractions of the stomach, but contractions of the abdominal, pelvic, and thoracic cavities. And so typically air is forced out at the same time as the food is forced out as well. So the epiglottis does not close typically during that. But that is why someone is not conscious, you do have to be worried about them aspirating from that vomit, then going down their airways. All right. Excellent. Here are the pretty pictures from your textbook. And again, we did that. And here are some pretty pictures from another textbook that I think does a nice job of showing it. And again, really does a nice job of emphasizing the trigger of it is that coming in contact with the arches, leading to the elevation of the larynx, which again closes the epiglottis and opens the upper esophageal sphincter. And then our larynx descends again, opening the airway, closing the foodway, and again, we see the movement of the uvula as well. All right, and that is the process of deglutition. Questions on that? All right, excellent. Like I said, the esophagus is primarily a muscular tube. That's function is just peristalsis. However, one thing we have mentioned, and here if we look, this is a, notice this is a cross section of it from a fairly proximal point, because the one thing we have talked about the esophagus is that the anatomy of the anterior or proximal 2/3, not anterior, proximal 2/3, and the anatomy of the distal third are different from each other. However, where they're primarily different is in the epithelial tissue. Notice and actually that's the one thing we should go back to. Let's do that. I don't know if we did this because it was at the end of last class. Oh, actually, we can just use this picture. I don't care. For the pharynx, right? We talked about let's start easy. We talked about the epithelial tissue that lines the oral cavity. What was the epithelial tissue that lines the oral cavity? Excellent. Non-keratinized stratified squamous epithelial tissue. So not surprisingly, what lines the oral pharynx?
Same.
Same. What lines the laryngopharynx?
Same.
Same. What lines the nasopharynx?
The same.
Nope, it turns out it's not. And if you think about it, that kind of makes sense, right? What passes through the oral pharynx?
Food, mucus, and air.
Food, drink, mucus, air, all of those things. Same thing for the laryngopharynx. But what passes through the nasopharynx?
Just mucus and air.
Just mucus and air. There's no food, there's no drink, so there shouldn't be sharp tortilla chips. There shouldn't be acidic coffee. There shouldn't be hot sauce or any of those types of things. So this is just dealing with air, which needs to be filtered, needs to be moistened, and mucus. So what type of tissue lines the nasal cavity? Come on, I know one of you is at least...
Keratinized.
No, not inside the nasal cavity. What's inside the nasal cavity? Come on, I know it's been a long time since you've had a chance to say it. I know you're all dying to say it.
Is it ciliated pseudostratified epithelial tissue?
There you go. Columnar. But absolutely, there you go. Ciliated pseudostratified columnar epithelial tissue. And that is also what lines the nasopharynx. The nasopharynx is also lined by a ciliated pseudostratified columnar epithelial tissue. All right, so again, it doesn't need to be protected from food and drink. So it's just about moving that mucus down towards the throat or down to the oral cavity so it can be swallowed or it can be spit out. Whereas the oropharynx and laryngopharynx need to deal with food, need to deal with drink. So they're lined with a non-keratinized stratified squamous epithelial tissue. And notice the same thing is true for the proximal part of the esophagus. Here we see a cross section through the esophagus, and we can see all the layers that we kind of expect to see for the most part. We have a mucosa. We have a submucosa. We have a two-layered muscularis, right? But let's look at this mucosa. What type of epithelial tissue is forming this mucosa? True, we do expect it to be columnar at some point, but remember we talked about how that columnar epithelial tissue starts from the distal third of the esophagus. I know this is a relatively low magnification, but hopefully you can tell this is pretty dense. And if you like, you can come over here and cheat based on what you see over here. What type of tissue do you think this is lining the proximal part of the esophagus? Izzy, you want to give us the letters again?
It's the non-keratinized stratified squamous.
Yeah, absolutely. It's a non-keratinized stratified squamous epithelial tissue. We have a submucosa. We have two layers of the muscularis. What type of tissue are these? The muscularis. What type of tissue is our muscularis?
Smooth muscle?
Smooth muscle, excellent. This inner layer, what is the orientation of this inner layer? Circular.
Circular.
What is the orientation of the outer layer?
Longitudinal.
Longitudinal. And what would be on the outside of this? Would it be an adventitia or would it be a serosa?
Adventitia?
Adventitia, absolutely. Remember, our esophagus is part of our sad puckers. So this is something that is held and anchored in place by a fibrous connective tissue in the posterior part of the thoracic and also abdominal pelvic cavity after it passes through the diaphragm. Excellent. So we see that nice cross section of it here. However, as we talked about, and as we know, about 2/3 of the way down, the epithelial tissue of the esophagus changes. And if you look closer at this at the histology over here on the side, you can see it is not a gradual transition. Right here where this arrow is, you can see how the esophagus is lined with a non-keratinized stratified squamous epithelial tissue, and then boom, suddenly it is lined with a simple columnar epithelial tissue. So that transition, which occurs about 2/3 of the way down the esophagus, is a rapid, dramatic transition. Excellent. What do you think this is?
Your diaphragm goes along there?
Not a bad guess. The diaphragm does have to pass through, although typically this transition doesn't necessarily occur where the diaphragm is. So that's not quite, but it's not a bad guess. No, this structure right here, this structure that I'm circling. Any idea what that might be? Doesn't look familiar to anybody? This is a cumulative class, right? Yeah, it's a lymphatic follicle. It's a MALT, a mucosa-associated lymphoid tissue. And there you go. So again, as we know, we're not surprised to find these just randomly thrown out within our mucous membranes. And that's a great example of one right there. All right, excellent. So we've seen the histology. We know the gross anatomy of our esophagus, and we've talked about what it does. And I think, again, not much to that. So any questions on the esophagus before we move on? All right, so here we are in the stomach. Again, the stomach is part of the major organs of the alimentary canal and has that same basic anatomy we expect, with a mucosa, a submucosa, a muscularis, and is the stomach retroperitoneal or intraperitoneal?
Intro.
Intra. So is it going to be lined with a serosa or with an adventitia?
Serosa.
Serosa, absolutely, excellent. So it has all of those basic components that we expect the basic anatomy to have. But there are also some specializations to the stomach. So while we start here at the gross anatomy, let's talk about some of the obvious specializations that we can see. The first is obviously a major enlargement of that tube. It isn't just a tube. We have this major enlargement that comes out of it, dividing this organ basically into four main regions. We have the proximal part of it, the part that connects to the esophagus, is the region known as the cardial region or cardia because of its close proximity to the heart. Right, this is why when you have an irritation of the stomach, we often refer to it as heartburn because of that referred pain. And I may have gotten a little aggressive with this, so let's cheat and grab it and move it a little bit more like that. This big, huge enlargement, the biggest enlargement of this is this, come on, that's what I want, thank you. Enlarged pouch-like structure known as the fundus. So this big, huge enlargement that reaches out, that sticks out, is the fundal region or fundus. It narrows at the end to a region called the pyloric region. And this funnel-shaped, highly muscularized pyloric region is where it funnels into the small intestine. And basically everything else in between is known as the body. So we have four distinct regions to this organ that can be seen with the gross anatomy, and we can also see some subtle differences between these regions when we look at it histologically as well. So when we look at these things histologically, we'll notice some differences there as well. So its enlargement is definitely one of the key features. We talked about this in the last class. We talked about a lot of stuff in the last class, so I'll remind you again. Notice the stomach has a concave curvature, which is known as the lesser curvature, and a convex curvature, which is known as the greater curvature. And remember, as we talked about, the lesser curvature is where the lesser omentum connects to the stomach from the liver, and the greater curvature is where the greater omentum comes off, draping down, and then connects to the transverse colon. However, the two other really big specializations that we can see at the gross anatomy view here, and the first is right here in the muscularis. Notice, as expected, we have an outer longitudinal layer. We have an inner circular layer, but notice there is a third layer, and the stomach is the only organ in the alimentary canal that has 3 layers to its muscularis. So when we look at it histologically, it's one of those things that easily jumps out and makes it very easy to identify, because it doesn't have 2, but actually 3 layers. It has the, again, outermost longitudinal, inside of that is circular, but the deepest, the one closest to the lumen, is an oblique layer. So basically we have 1 longitudinal layer, 1 circular layer, and 1 oblique layer, giving us 3 layers to the muscularis externa. Why is that important here?
Different directions to contract, to be able to move the food to digest.
Yeah, remember, muscle's a one-trick pony. Basically, when it contracts, it gets smaller. We know circular layers constricts the organ, longitudinal shortens the organ, and this oblique twists it. So basically, by having more layers, more muscles at more orientations, right, it allows for more motility of the stomach, more churning for the stomach. Because like I said, I know every single person in this class chews their food the 40 times they're supposed to before you swallow it down. But some people take a big, huge bite, chew twice, and then chuck it down into their stomach. And so that churning of the stomach, and that's really the advantage of these 3 layers, we get much more elaborate mechanical movement of the food that we call churning. And so that is going to be important for the processing of the of our food, of our now bolus. The other gross anatomy structure that you can see is, notice as we look at the inner surface of the stomach, the mucosa and submucosa, and this is important to point out. There are these massive folds in the stomach known as rugae. These rugae are folds in both the mucosa and submucosa. So there are these big, huge, massive folds in the mucosa and submucosa. And what is their function?
Allow it to expand?
Yeah, absolutely. Right. The 4th of July is right around the corner. Right. Our president has assured us that we'll be able to have a normal 4th of July, and hopefully that'll be the case, because on the 4th of July is when the greatest sporting event of all humanity takes place.
Hot dog eating contest.
There you go. Absolutely. Joey Chestnut going for the record yet again. Right. And I think last year he didn't quite reach the record and only ate something like 62 hot dogs and buns in 12 minutes. Right? If you think about the ability of the stomach to be able to expand to accommodate 62 hot dogs and buns is a truly remarkable event. And so absolutely, that ability of those rugae allow for the massive expansion. Your stomach, when empty, is about the size and shape of your 2 fists. So if you make your 2 fists and put them together, that is the size of your stomach when it is empty. And if you think of, okay, maybe you don't eat hot dogs on the 4th of July, but a little bit after that, we hit Thanksgiving. Is the amount of food on your Thanksgiving plate just 2 fists worth of food? No, absolutely not, right? So that ability to be able to expand, to handle that 3rd serving of stuffing and that 2nd piece of pecan pie, right? We need that ability to be able to expand the stomach, and those rugae allow us to be able to do that. All right, excellent. That is our gross anatomy specializations. Notice here, I love this picture. Notice here we can see the rugae with a lower magnification view. And again, notice we can tell these are rugae because what's this line right here? Anybody know what this line right here is? All right, that is the Muscularis Mucosa, right? We have our mucosa, our lamina propria, our Muscularis Mucosa, and these 3 together form our mucosa. Then we have the relar tissue that makes up our submucosa. Notice here's our muscularis. So notice these big, huge folds are comprised of both the mucosa and the submucosa. They allow for that expansion. And notice the muscle doesn't conform to those folds in the same way. So these rugae are expansions of both the mucosa and the submucosa. Of course, this is the muscularis, and then the dark line on the outer surface would be the serosa. We can see that really nicely in this view of the stomach. However, if we look closely, microscopically at just the mucosa, and really that's the key here, now we are just in the mucosa. And again, we can tell that by finding that muscularis mucosa. Here's the muscularis mucosa. Remember, that little bit of smooth muscle is what separates the mucosa from the submucosa. Right? So notice just in the mucosa, we get this additional specialization to the stomach, what are known as the gastric pits and the gastric glands. Notice the surface of the stomach, the surface of the mucosa of the stomach is not smooth. There are these massive invaginations, and these massive invaginations are called the gastric pits. However, notice these pits aren't just hollow at the bottom. Down inside of these gastric pits are some highly specialized cells, and these highly specialized cells are what are known as the gastric glands, or form, I should say, the gastric glands. And of course, those gastric glands produce our gastric juices. So the pits, the indentations are the gastric pits, and then deep to that is the gastric glands. If we look a little closer, clear that, come here. Here we go. So again, notice we can see that simple columnar epithelial tissue that is lining the surface of, I'm not sure what that is, so I will, well, someone's vacuuming or whatever, we'll mute that. We have our simple columnar cells lining the surface of the stomach, and notice they also line the gastric pits. We're going to do this in just a second, but I will remind you also there are massive numbers of goblet cells located in this epithelial tissue that is lining the surface. And someone remind me again what goblet cells do?
Produce mucin.
Exactly, they produce mucin, a protein, and that in this case, these goblet cells lining the surface of the stomach produce a very thick and alkaline mucus. Why might we want a thick and alkaline mucus on the surface of our stomach?
Protect the layers from the acid inside of it.
Yeah, remember, as we talked about, the gastric juices that are being produced by the stomach have a pH of between 1 and 2. Right, and we know cells, which are chock filled with protein, right, can be damaged and denatured by that. So we need this thick, sticky, alkaline mucus to line the surface of the stomach to provide that protection. And so we have this normal, simple columnar epithelial tissue with those goblet cells producing that thick, sticky, alkaline mucus. However, deep inside those pits, we have our gastric glands. And this gastric glands contain some highly specialized cells that, again, are responsible for producing our gastric juice. And as you can see by the illustration here, and we'll talk about them in just a moment, there are 4 main types of specialized cells found in this gastric gland. They are the mucus neck cells, that are not the same thing as the goblet cells of the normal epithelial tissue, parietal cells, chief cells, and enteroendocrine cells. I want you to notice one more thing about this illustration. Notice that while these 4 cells make up our gastric gland, I want you to notice that the mucous neck cells are typically just located at the proximal portion near the entrance to the gland. Whereas I want you to notice that the enteroendocrines are the deepest cells at the base of the gland. Let's talk about these a little bit more. Here we see another illustration that again shows these. Notice we have our normal surface epithelial cells with those surface mucus cells producing that sticky alkaline mucus that is going to, again, protect our stomach, right? If we're not producing enough of this sticky alkaline mucus, or if we start overproducing acid, where it starts eating away at this mucus too much, we can start causing damage to the epithelial tissue and the lining of the stomach. And what do we call that condition when it occurs? An ulcer, absolutely, right? If that ulcer goes down deep enough, like all the way down to the submucosa, we can actually get a bleeding ulcer as a result of that as well. So that is definitely a bad thing that we don't want to have happen. So having that surface mucus cells producing that sticky alkaline mucus is very, very important. But remember, these other cells are the ones that are found in the gastric gland. And again, in something that I'll be saying a lot in this section, obviously it depends on when you eat, how much you eat, how often you eat, the composition of the food you eat. All of those things can cause this to vary, but on average, a person produces about 1.5 liters of gastric juice during the course of a day. Now, let's talk about the cells that are producing this. Starting first with the parietal cells. I am not going to hold you responsible for distinguishing the 4 cells of the gastric gland histologically. So I'm not going to show you a histology slide of a gastric gland, point at one cell, and ask you to identify what it is. However, there are some things on a model like this or an illustration like this that can be easy to identify. One of the easiest are the parietal cells, because these parietal cells kind of have this fork-like appearance to them, where they have a large centrally located cell body and these large processes that stick out, giving it this kind of bifurcated or pronged type of appearance. Makes it very easy to identify on an illustration. It looks that way histologically as well, but honestly, it's a little harder to distinguish, and I won't hold you towards that. But an illustration or a model or something like that, sure, you should be able to recognize that as a parietal cell. This parietal cell is what produces our hydrochloric acid. Now, my big question for you is, why? Why do we need this hydrochloric acid? See, this is the part where I ask the question and then you guys answer it.
Break down food.
Okay, how? You are absolutely correct. But how does hydrochloric acid break down food? Let's be more specific. What type of food? All foods?
Proteins?
Proteins, excellent. Proteins, absolutely. Primarily is used for the breakdown of proteins. Why? What's so special about proteins?
Harder to break down than carbohydrates?
Okay. Why?
They have to be broken down in more steps.
Okay, there can be more steps in it. Sure, that helps. So how does hydrochloric acid help in this?
Breaks more bonds?
And that's excellent. So good. I'm glad we're doing this, because I'm glad I asked this question. There you go. That was the term I was looking for. So we got to be careful. It does not break bonds. In breaking down food, hydrochloric acid does not break, well, let's say it this way, does not break peptide bonds. Remember, peptide bonds are what hold amino acids together. But what kind of bonds does the hydrochloric acid disrupt? I know you guys know the answer to this. You guys are just overthinking it. All right, Roe came up with the right answer. It denatures proteins. What hydrochloric acid does is it denatures proteins. But what does that mean? Not ionic bonds. We don't have ionic bonds in our proteins, but what do we have? Well, so if it's not covalent bonds, peptide bonds, if it's not ionic bonds, then what are we left with?
Covalent?
Nope. Hydrogen bonds. All right. Remember, proteins have their shape because of the hydrogen bonds that are giving the secondary and tertiary shapes to them. Right, but those hydrogen bonds give them the secondary shapes, which causes the tertiary shape and gives it its shape, gives it its function. Changes in temperature, changes in pH disrupt those hydrogen bonds. And when we disrupt those hydrogen bonds, we denature the proteins. We take it from a coiled up structure and basically unwind it, change its shape. Notice we're not breaking bonds. This is not a chemical breakdown. This is actually a mechanical breakdown. So this is mechanical digestion. It opens the proteins up, making it easier for enzymes to get in there and cut the bonds. So hydrochloric acid doesn't actually break any of those peptide bonds. It doesn't separate any of the amino acids from that protein. But it does denature the protein, opens the protein up, making it easier for the enzymes to get in there and do its work. So technically, it is changing the shape without changing the composition. So technically, hydrochloric acid actually denatures the proteins, which is a form of mechanical digestion. Just like chewing makes it easier, increases the surface area, makes it easier for the enzymes to do the work, denaturing makes it easy to do the work. All right, excellent. Now, notice all these things we've been talking about sound really important, right? You got to store those 62 hot dogs that you have on the 4th of July. You need to, and of course, the other function of this hydrochloric acid, as we've talked about, is protection, right? It was part of our first line of defense, stopping pathogens from getting into our body. So it's denaturing proteins. It's protecting us from pathogens, right? It's storing those 62 hot dogs. It's churning them if you didn't chew the food enough. It makes it sound like the stomach is a pretty vitally important structure, right? You can't live without a stomach. Stomach is definitely the most important organ in the alimentary canal, right?
You can live without a stomach.
Absolutely, you can live without a stomach, right? The stomach definitely has rock star status, right? If you grab some random person on the street and ask them for an organ in your alimentary canal, most people will probably say stomach. But there is only one thing your stomach does that you can't live without, right? Everything it does definitely helps in digestion, storing the food, doling it out a little bit of time to the small intestine, right? Churning the food, denaturing the food, all of those things help, but none of it is vital for your existence. So absolutely, as Allison pointed out, you can absolutely positively live without a stomach. Have it ripped out of your body if you need to, right? We talked about those bleeding ulcers, or if you have cancer or something like that, they can rip it out and connect your esophagus straight to your small intestine, and you'll be just fine, except for one thing. The one thing that your stomach does that you cannot live without actually is done by these parietal cells. These parietal cells not only produce hydrochloric acid, but they also produce a substance called intrinsic Intrinsic factor is a special chemical composition that basically allows your large intestine to absorb vitamin B12. Well, that doesn't sound like too big of a deal until you learn what vitamin B12 does. Vitamin B12 allows you to make red blood cells, right? Remember way back in ancient times when we were studying the cardiovascular system, we talked about how red blood cells only last for about 100 to 120 days. So it is vitally important that we keep making red blood cells to replace all the ones that die. And to do that, we have to have B12. And so the one thing your stomach does that you can't live without is this intrinsic factor. You can drink a whole bucket of vitamin B12, but if you don't have intrinsic factor, your large intestine is not able to absorb that in your body and not able to use it. So, as Allison pointed out, if someone has their stomach taken out because of cancer or ulcers or something along those lines, they're getting B12 injections for the rest of their life because they're not going to be able to absorb it from their diet anymore. Right? But that's it. Well, it is a very, very helpful organ, but this right here is the only thing it does that is vital for our existence. Now, as I mentioned, our parietal cells denature proteins, allowing them to be able to be more easily broken down by enzymes. So it shouldn't surprise you that chief cells produce an enzyme that helps us to break down proteins. It's another thing we should be thinking about. Not only are we talking about the different iterations of food, again, we've gone from food to bolus, and we're continuing that process, but we should also start thinking about when the chemical digestion of our macromolecules occur. Remember, our chemical digestion of carbohydrates, where did that begin? Where did we start?
In the mouth.
Yeah, in the mouth with that salivary amylase. Well, now here in the stomach, we also get the beginning of protein chemical digestion, and it occurs by a special enzyme called pepsin. Pepsin is an enzyme that breaks down proteins. Now, aren't cells chock filled with proteins? This is the part where you say yes emphatically.
Yes.
Excellent.
Yes.
Absolutely. Thank you. Thank you. Excellent. Absolutely. So if you were a cell making pepsin, you got to be really, really careful because you don't want that enzyme chopping up your proteins. So not surprisingly, these chief cells produce it in an inactive form as pepsinogen. And so it is released in that inactive form. And one of the things that activates it is the hydrochloric acid. Hydrochloric acid can activate pepsinogen into pepsin. And then, of course, once you have pepsin, since pepsinogen is a protein, it can then even activate even more. And I think I actually have a pretty picture that shows this. Here we go. Yep. So here we go. Our parietal cells producing the hydrochloric acid that helps to convert pepsinogen into pepsin. And then, of course, pepsin can help to facilitate that process, making even more pepsin. And then, of course, that pepsin can break down proteins in our diet. All right, so again, it's kind of a positive feedback process here to ramp up digestion very, very quickly when food is presented to the stomach and the stomach starts producing this gastric juice. Now, in you and I and everybody else that's hanging around listening to this right now, these chief cells primarily just produce pepsinogen. However, for those of you who have little kids, especially infants, these chief cells also produce two other enzymes. One is renin, which helps to break down milk sugars, And the other is gastric lipase, which helps to break down milk fats. And why might it be important for infants to be able to produce additional enzymes that help to break down milk sugars and milk fats?
Milk is their entire diet.
Exactly, absolutely. Milk is their entire diet. So having those additional things help. Notice we do not continue to produce these throughout our entire life. So one of the things that happens to us as we age is we do become more milk intolerant as we age. Our ability to be able to process milk sugars and milk fat decreases as we age. It happens to everybody. Some people it happens more radically than others. Some people become more lactose intolerant much earlier than others do. But we all do decrease in our ability to process milk sugars and milk fats as we age. All right, questions on that? All right, excellent. So we've talked about those two. There are those, and those are kind of mixed in throughout the middle of this gland. But remember we talked about these mucous neck cells. What's interesting about these mucous neck cells is that they also produce mucus, as their name would indicate, but it is an acidic type of mucus. And the function of this acidic mucus is not fully understood. It is believed that it also may play a role in helping to activate the pepsinogen into pepsin to help in the breakdown of our food. The other thought is that it may play a role in helping to stop the gastric gland from becoming congested. If you think about it, if our gastric pit and its surface mucous cells are producing a massive amount of sticky, thick, alkaline mucus, that sticky, thick, alkaline mucus may congest the pore of our gastric gland and stopping the gastric juices from being released. So they think that maybe this acidic mucus helps to break up the thick alkaline mucus and keep the opening clear. But like I said, the function of it is not fully appreciated and understood. We know they're there. We know they produce an acidic mucus, but what exactly that acidic mucus is doing is not fully understood. And that brings us to our last cell. That last cell, remember, is that enteroendocrine cell located way down deep at the base of the gastric gland. Why is important? Because as the name indicates, enteroendocrine cell means that it is an endocrine cell. And what does it produce? What do endocrine cells produce?
Hormones?
Hormones, exactly. And if this gland is, if this cell is a single-celled gland producing hormones, do we want to make those hormones and then release them into the gastric pit and out into the lumen of the stomach? Is that what we do with hormones? Secrete them outside of our body? These are the easy questions.
No.
No. So what we want to do instead is release it into the tissue, into the interstitial fluid, where it's going to get picked up by the blood supply, and it is going to be circulated throughout the body. So it kind of makes sense that it's in the deepest part of the gland because it doesn't need to be releasing it out of the gland. It wants to release it into the submucosa, into the mucosa, into the capillaries, get picked up and circulated throughout the body. Gastrin is a very important hormone that plays an important role in enhancing the activity of the stomach. It basically does two things. It increases motility, making the stomach churn more and more vigorously, and it increases production of the gastric juice. So we make more pepsin, we make more hydrochloric acid. Now, of course, this is a circulating hormone and it is going to affect other organs as well. But for right now, we'll just focus on its function for the stomach. And that function for the stomach includes increasing motility, increasing production of gastric juice. So it increases the overall activity of the stomach. It enhances the activity of the stomach. Questions on that? All righty. Controlling the activity of the stomach is going to be important. Excellent. Let's look at this histologically first before we get into this, though. Notice here we can see the mucosa. And again, one of the first things you should go looking for is that muscularis mucosa. Here's the muscularis mucosa, that smooth muscle layer. So that tells us this is the submucosa down here. And that means all of this is our mucosa. And with a mucosa this thick, that is a dead giveaway you're looking at the stomach. We can see it is lined on the surface by simple columnar cells, but we can see these big, huge, massive glands that are penetrating down deep into the mucosa, and these are indeed our gastric glands. And notice if you look closely, some of these cells are darker than others. The shapes of them are going to be different. But like I said, it is possible to tell the mucous neck cells and the parietal cells and the chief cells and the enteroendocrine cells, but I don't expect you to be able to do it. So just recognizing this, and you can clearly see, while I don't care that you're able to distinguish the individual cells, you can clearly tell the difference between the gastric pit, which is lined with the normal simple columnar cells, and the gastric glands, which are this part here. So notice the pit ends there, the pit ends there, the pit ends here. So basically we can kind of see the line of where the pits end and the gastric glands begin. So good, strong giveaway that this is the stomach. Now, there are some differences. And again, here's another nice example of this as well. Notice we can again see the simple columnar cells and then the pits coming down from them. This is what I wanted. So let's start here. Notice here, we've got lots of different shades to this. This is typically what the fundus, fundus, or the body of the stomach would look like, with the large, deep glands, with lots of parietal, lots of chief cells, lots of things along those lines. However, notice down here, this is more the pyloric region. In the pyloric region, notice when we look at the glands here, much, much more goblet cells. The pyloric region of the stomach basically is involved in moving the food into the small intestine. So it has a very, very thick muscularis. Notice we can very clearly see the three layers. Layer 1, layer 2, and layer 3. Of course, this first layer, what layer would that be? What's layer 1?
No.
I'm sorry?
From the inside or the outside?
Well, here's the lumen, right? So the one I've labeled as 1, what would that be?
Oblique.
Yeah, it's the oblique. Excellent. Number 2 would be
Longitudinal.
longitudinal? Or is 3 longitudinal?
3 is longitudinal.
So then that makes 2 circular. There you go. Excellent. So oblique, circular, and longitudinal. Notice we can clearly see that the cells are oriented at different orientations, but what orientation they are doesn't really matter. We can just clearly see the boundaries between the changes of their shape, and that makes it very easy to distinguish those 3 layers. So seeing 3 layers, dead giveaway you're in a stomach. When the 3 layers are very, very thick like this, and you see that the gastric glands are primarily lined with goblet cells, then you know that you're in the pyloric region. So I expect you to be able to distinguish the pyloric region from the rest of the stomach. where it's not as muscular, and the glands are much more varied in the gastric glands. So again, that is something I expect you to be able to do histologically. The one other cool thing I wanted to show you, and this is actually, as you can see, we are going from the stomach to the small intestine. At the end of the stomach, the distal part of the stomach, the pyloric region, which has that big, huge muscular layer to begin with, notice the circular layer of it comes up and enlarges, becoming this big, huge, massive structure known as the pyloric sphincter. And the pyloric sphincter is what controls the movement of food from the stomach to the small intestine. So we see this big, huge, massive enlargement of the circular layer, forming this big, huge, thick muscular valve that is going to control the movement of food from the stomach into the small intestine. So our stomach is bounded on both ends, just like our esophagus. We have the lower esophageal sphincter, or the cardioesophageal sphincter, gastroesophageal sphincter at the proximal end of the stomach. And at the end, we have this big, huge, massive pyloric sphincter. Now, again, you have that loving 2-year-old who, again, starts eating that cheeseburger and decides they don't want it anymore. So they spit it out into your hand to give it to you instead. And again, as we've talked about, we have that big change in iteration from that food to the bolus as it is masticated and mixed with saliva. And again, that occurs in the mouth. But here inside the stomach, that again, so again, instead, if you think of it in terms of that 6-month-old who you haven't burped properly, so the milk goes down, and then shortly after, the milk comes back up. When the milk comes back up, does it smell and feel the same as it did when it went down? No, because when it reaches the stomach, what happens is we mix it with that gastric juice, we churn it with the muscles of the stomach, and we form a substance we call chyme. So we have our second iteration of the food, bolus that occurred in the mouth, and then it is converted into chyme in the stomach by mixing it with the gastric juices, by churning it, continuing the mechanical breakdown. Again, protein digestion begins thanks to pepsin. And we should also, here under mechanical digestion, we should say specifically for proteins that this is also denaturing with the hydrochloric acid. Protein digestion begins via pepsin. Notice also there's going to be a little bit of fat digestion that is going to occur here in the stomach. From what again? What's the enzyme that's going to cause the breakdown of fat here in the stomach?
Leptin?
Nope, not leptin, although it's close. It's definitely something lipase, right? It is lipase. Where did we have a lipase that we made? True, we are going to make it to the pancreas, but the pancreas doesn't feed into the stomach. The liver does produce bile, but it doesn't feed it into the stomach. How did the stomach get some lipase? It clearly didn't make it.
Adipose tissue?
Was there something in the oral cavity that we said made a lipase?
Lingual.
There we go. Remember, as we mentioned, our tongue produces a lingual lipase, which remember we said was inactive in the oral cavity. Right? You can't put that chunk of fat on your tongue and have it dissolve and be broken down in your oral cavity. But when you swallow it down, that lingual lipase becomes active and starts the breakdown of fats in the stomach as well. It's only a little bit, but it's there. And obviously, remember also, if you were a infant, you would have a lipase being produced by a gastric lipase being produced by your chief cells that would help you break down mom's milk. And then, of course, the parietal cells produce that hydrochloric acid. And we talked about the mucus cells and their function. So again, we've talked about a lot of the physiology of the stomach already. So what we need to do is talk about how we're going to control it. Actually, I guess before we'll talk about the other stomach. There is some limited absorption that does take place in the stomach, very, very limited. Alcohols, as many of you know, you don't want to drink on an empty stomach. Medicines like aspirins, other short chain fatty acids, things along those lines, electrolytes, and water. And one of the interesting things about water is cold water is actually absorbed more rapidly than warm or room temperature water. I don't actually know why that is, but after a workout, it's actually better to drink ice cold water than it is just room temperature water because it is actually absorbed by the stomach more quickly. I don't happen to know why. And like I said, we want to churn that chyme and present it to the small intestine. There are these Peristalic waves that occur across the surface of the stomach all the time, right? Unless you're eating right now, your stomach is slowly churning away at a rate of about 3 per minute. They're actually pacemaker cells similar to the cells that we've talked about in the heart, that are actually able to produce an action potential, and that action potential spreads across it. It's part of that enteric plexus we talked about. And the pacemaker cells are actually called cells of Cajal. Ramon E. Cajal was a very, very famous neuroanatomist who was a beautiful artist. And he loved to look in the microscope and do these beautiful drawings of the things that he would see under the microscope. And he was an absolute anatomy whore and put his name on everything he drew. But again, truly amazing. His life story, if you're ever bored, look up Romain E. Cajal. He is an amazing individual. But these produce that rhythm. And of course, then the gastrin and our neural input enhances that when we are dealing with food. Again, those contractions become most intense in the pyloric region, where those powerful contractions are churning the food and then also expressing a small amount of it into the small intestine to be processed. Again, it depends on the volume of food you eat, the composition of the food you eat, and the timing of the food that you eat. But in general, when food is presented to the stomach, a meal is presented to the stomach, it empties in about 2 to 4 hours. All right. So those are the functions of the stomach. What we need to talk about next is how we control the stomach. Now, one of the points I want to emphasize is many of the mechanisms we are going to talk about influence other organs in the alimentary canal as well. However, when we talk about regulation of gastric activity, or you see an essay question about regulation of gastric activity, we are just talking about the stomach. So we just want to focus on the stomach. And as you notice, there are 3 main phases of control for the stomach. And as we'll see, and based on the names, they are basically associated with where the food is. Where the food is influences our control of the stomach. And so this is, I won't say one of the denser, but there is a lot of material we need to cover on this. And so I want to make sure we come at it fresh. We've been talking for a while now. So let's go ahead before we dive into this and take our first break. Looks like it's 9:15 right now. So let's restart at 9:30. And at 9:30, I will start the recording and we will pick up from here. So when we come back, we're going to talk specifically about how we control the stomach, a regulation of gastric activity. So I will see you in 15 minutes. All righty. So I like this illustration because I think it does a nice job of emphasizing the points of the things that we're going to talk about. Our goal is to talk about control of the stomach, gastric activity. And it, as we talked about, it occurs in 3 phases. And those 3 phases are totally dependent on where the food is located. First is the cephalic phase. Cephalic, of course, means what? Head. So this either means obviously when it's in your mouth because you've tasted it, but it could also be seeing the food. It could also be hearing the can opener opening or smelling it or something head related. The key is this is what happens before the food reaches the stomach, right? Because again, if we're going to be efficient in working the stomach, we want to prime the pump. So this is occurring before the food reaches the stomach. Obviously, the gastric phase, coffee and Bailey's, that works too. Okay, that works too. Coffee and Bailey's. Plus, that Guinness is really hard to make green, so that's the other challenge as well. Gastric phase is when the food is obviously in the stomach. So when the food is in the stomach, obviously we want to have some control over what's going on in the stomach once the food is there. And then lastly, as the food leaves the stomach into the small intestine, right? We want to be able to control it. Remember, we talked about the stomach has rock star status in that everybody knows the stomach. But remember, we also talked about there's only one thing that it does that is vital. The true workhorse organ of the alimentary canal is the small intestine. It is where the majority of the work takes place. And so we want, once food starts entering the small intestine, we want to be able to control the rate at which the stomach presents it to the small intestine, so the small intestine can be as efficient as possible. So again, where the food is going to determine how we control the stomach. The other thing that this illustration shows nicely is that there can be both excitatory and inhibitory inputs that are going to influence this and affect this. So let's work our way through this. Let's start first on the whiteboard. I think this could be something that might be worthwhile to do on the whiteboard. So, because again, I think sometimes doing it on the whiteboard allows us to more easily organize our thoughts as we go through this. So step 1 is, like I said, the cephalic phase. And this, oops, I spelled phase correctly. And again, this is basically while the food is in our head. So this is again before the food reaches the stomach. So this is going to involve sight, right, smell, taste, or even just thinking about it, right? If you really, really think back to your last anniversary and that amazing meal that you went out to, all right, and the dim sum or whatever it was that you had, I'm sure you could think about some food. And if you really sat here and thought about it long enough, right, you'd probably be able to affect your digestive system in that fashion. Now, as we know, there are 2 ways to control an organ like the stomach. The cephalic phase is going to be controlled primarily by the endocrine system or the nervous system. What do you think for this one here?
Nervous?
Absolutely. This is going to be nervous system control. Absolutely. And is this going to be excitatory or inhibitory?
Excitatory.
Absolutely. So again, that nice, someone comes up and presents you with this beautifully, barely, still blue tri-tip, sliced open with lots of juices pouring out of that. And everybody's stomach is going to start exciting. Everybody's going to start salivating in response to that. Is there anybody who's not going to respond that way to that big, juicy hunk of meat?
The vegetarians.
Yeah, exactly. So notice if there's a vegetarian out there, they may not necessarily respond to that the same way. Or even if you do like that tri-tip, as someone, excuse me, is walking across the lawn to present that to you, and they trip and drop it, and it lands in dog poop, and they stick it up and slap it on the plate and still give it to you. Are you going to start salivating as a result of that as well? No, probably not, right? So notice this cephalic phase can be excitatory or inhibitory. And a lot of that has to do with your own personal preferences, right? This is a very personal type of response, right? That little kid, you may love mushrooms. I love roasted mushrooms, right? Roasted mushrooms are amazing. I love the smell. I love the taste. I like they're amazing. I have a daughter who hates it. So while I smell that roasted mushroom, or I look at it on the plate, or I taste it in my mouth, that's obviously going to be excitatory for me, right? Her, I have to shove it in her mouth, and I have to pinch her nose and keep my hand over the top of her mouth to get her to swallow that down. Aside from the fact that she doesn't like the taste, the smell, or the look of it, the fact that I'm pinching her nose and have my hand over her mouth, is that necessarily going to lead to excitation of her stomach in anticipation of that food? No, absolutely not. So notice in this case, we have this neural effect, and it can be excitatory or inhibitory. Now, obviously, the goal is to eat things that you want to eat. So obviously, we want this to be excitatory, right? But it is important to emphasize that it can be inhibitory in some instances. But our goal is to prime the pump. The goal is to get the stomach ready for food, because that anticipation of the food is on the way. And so primarily what starts to happen in this is this neural input. And again, let's skip this and move this down a little bit. If we're going to be doing excitatory, what would the neural control, what would the pathway be for excitatory? Or not pathway, I won't hold you for pathway, but what branch of the nervous system would be responsible for the excitatory?
Sympathetic.
Sympathetic for excitatory in this case?
Be the parasympathetic.
Parasympathetic.
Parasympathetic. Pathetic. Right, absolutely. So our excitatory is going to be parasympathetic, whereas our inhibitory would be sympathetic. Pathetic. Excellent. So our goal is to get the stomach ready. So using the parasympathetic nervous system, we are going to primarily stimulate, so I'll put in parentheses because again, our goal is to prime, to stimulate primarily an increase in gastric secretions, starting the production of our gastric juice as a result of that. And if the anticipation becomes long enough, then it also can increase motility as well. So our stomach can start churning in anticipation. You get that gurgling in your stomach and that anticipation of the food coming in. Conversely, if you're stressed because you know that a plate of mushrooms is sitting in front of you and you're going to be forced to eat it, right? You can get a decrease in the activity, a decrease in the glandular secretions, all that kind of stuff. And again, this is happening in other areas as well. We are focusing, remember, this is gastric control. Gastric regulation. Other things are happening as well. Obviously, that anticipation of your food, for instance, gets your saliva going, right? So we're not saying that other things aren't happening in other organs. Absolutely, other things are happening in other organs. Right now, we're just focusing on what's happening in the stomach. So our goal, assuming we're eating something we want to eat, is to increase gastric secretions, increase motility, using our parasympathetic nervous system to get the stomach ready for food. But again, it is a very personal response. Stress, right? Something having to be food that you don't want to eat, any of those types of things are all things that could cause an inhibition of this process as well. All right, excellent. So we put it in our mouth, we masticate it the 45 times we're supposed to before we swallow it down, and then it reaches our stomach and we get to the gastric phase. All right, this is when the food reaches the stomach. All right. All right. When that food reaches the stomach, basically what we get are local cues primarily. Does your stomach care that you had to bribe your kid with 20 bucks to eat 3 Brussels sprouts? No, all it knows is that it is stretching because food is being presented to it. So in this case, we're getting a stretch of the stomach. We are getting a change in pH because making you have to bribe your kid depresses you, so you're drinking your wine as a result of it, or the presence of certain substrates, fatty acids or particular amino acids or things along those lines can be present. All of these local cues, whether they're mechanical or chemical, and that's really the key, they're either mechanical, chemical, or it could be related to the osmolarity of the food, is going to be a local cue. And that local cue is going to have a local effect. What does that sound an awful lot like? Local cue, local effect, local processing.
The intrinsic plexus.
Yeah, it sounds like that intrinsic plexus, like that local reflex, that short reflex. So not surprisingly, there is a nervous system control. And in this case, the nervous system control is going to be that enteric plexus. And is this going to be excitatory or inhibitory?
Excitatory.
Excitatory. Right, so this is going to be an excitatory effect from that enteric plexus with that local cue and that local effect. Of course, that local effect is going to be to increase motility of the, oops, I spelled motility, right? Of the muscularis, right? So we get more churning. And increase secretions from our gastric gland. Of course, we know that increase in secretion from the gastric gland includes hydrochloric acid, includes pepsinogen, but it also includes the release of gastrin. Enteroendocrine cells, and again, these particular enteroendocrine cells are sometimes called G cells because they're the ones that release gastrin. Gastrin, remember, is a circulating hormone that affects the stomach. So notice in this case, it is nervous system control and hormonal control. Our enteric plexus, which is excitatory, and gastrin, which remember is also excitatory. So this second phase, again, is that gastric phase. This is when the food hits the stomach. And again, it's not so much about the motivation of how the food got there or what the food is, right? If you're on Survivor or what was the show that Joe Rogan did before he did all the MMA stuff? Fear Factor, right? Or something like that, where you have to eat a bug, right? You may not have enjoyed the thought of putting the bug down, but once the bug hits your stomach is going to be churning it up, right? And so again, this is primarily excitatory in its function because it's with dealing with the food that's in there. However, if you really were on Fear Factor and you had to eat that beetle, and in your haste to get it out of your mouth, you maybe didn't chew it too much and just swallowed it straight down, and you could actually feel those bugs' feet going along inside of your stomach, would that necessarily be a pleasant experience that you think might increase your enjoyment of that meal?
Yep.
Okay, maybe for some people, but not necessarily for others. Or if you really did have to pinch the nose and hold somebody's mouth to get them to swallow something. With stress, with too much pH, if there's too much acid, right, things along those lines, this can become inhibitory. And if it becomes inhibitory enough, as we talked about, that can actually induce vomiting, right, as a result of that. But again, this is not the way it is typically going to occur, right? This is not what typically happens. But again, in extreme stress, this can, or if it's between too acid, or if the food is soured in some way or something like that, spoiled in some way, in those types of really stressful types of situation, this can be inhibitory, and that can ultimately lead to the vomiting of the material as a result of that. So it can be inhibitory in extreme situations, but it is primarily, this one is primarily, right, excitatory in nature. All right. Questions on that? All right, excellent. That brings us to our third phase. The other thing that is occurring during this gastric phase is that food is being presented from the pyloric region into the small intestine. And that leads us to the intestinal phase. So in the intestinal phase, this is when our food is presented to the small intestine. And the small intestine needs to do the work. Remember, as we talked about, our small intestine is the workhorse of our alimentary canal. And in my mind, this is the part where I always envision that very famous I Love Lucy, where she's sitting there working at the chocolate factory, and the chocolates are coming down the conveyor belt, and it's her job to put them inside the wrappers so that they can go into the box. And at first she's doing fine, and then suddenly more and more and more and more start coming faster and faster and faster and faster, to the point where she's overwhelmed and she can't do the work efficiently, right? And while that's fun in a TV show, that isn't fun for the small intestine. And so our goal here in the small intestine is to regulate the rate of presentation of food, and more specifically presentation of chyme, to the small intestine for maximal efficiency. Towards that end, this process is going to be both excitatory and inhibitory. Oops. It's going to be excitatory first, but only briefly. And then it is going to be inhibitory second. And when it comes to how we control this, it is also going to be both neural and hormonal. So let's start first with the excitation. When food is first presented to the small intestine has its own enteroendocrine cells. And these enteroendocrine cells release intestinal gastrin. Intestinal gastrin is identical to stomach gastrin, with the only exception being where it's produced. However, both gastrin and intestinal gastrin end up in the blood, and when it gets to the muscularis of the stomach, when it gets to the gastrin gland of the stomach, does that gastric gland, does that muscularis care whether the gastrin was produced by the stomach or by the small intestine? No, it's going to affect it the same. So here we have hormonal control that is excitatory. It stimulates more secretion and more churning. Excellent. There's food in the stomach. Get that food moving on to our small intestine so we can process it. So the first effect, and it's a brief effect, is going to be excitatory. However, like we've talked about a lot of processes in the body, fast is good, but faster isn't necessarily better. And that's going to be the case here, right? What's going to happen next is that we want to modify the rate of processing and here for the first time, we want to do it based on the composition of the food. Notice when your stomach stretched in the gastric phase, did it care whether it was being stretched by pork or whether it was being stretched by spaghetti or whether it was being stretched by buttermilk? No, it didn't care. It just stretched and it increased the rate of churning, the rate of production. However, here for the first time, we actually care about the composition of food. Because as someone mentioned earlier, is it equally easy to break down carbohydrates, to break down proteins, and to break down lipids? No. Which of those three are the easiest to break down?
Carbs.
Carbohydrates, absolutely. Which of those three is the hardest to break down?
Protein.
No, actually, the lipids are actually harder to break down than proteins. So let's say it's carbs, then proteins, and then lipids. Lipids are actually the most challenging for the body to break down. So in this case, the rate, if you had a very carb-rich meal versus a very lipid-rich meal, you would want to modify the rate at which that was presented so that those lipids were presented more slowly to the small intestine so that we can modify the rate. This is going to occur both neurally and hormonally. Oops, I guess this doesn't need to be in capitals anymore. And in most cases, the goal here is going to be to slow the processing of the stomach and slow the presentation of the food to the small intestine. Run out of room, so let's go back to the pretty picture and look at this here, and we'll finish this off.
Where did you say the enterocrine cells are located?
In this case, it would be in the small intestine. So there are, remember, enteroendocrine cells are individual cells located within the wall of the mucous membrane that produce hormones. We first ran into them at the base of the gastric glands. That's where the G cells, those specific enteroendocrine cells, produce and release gastrin. But there are also, as we'll see when we talk about our small intestine, dozens of enteroendocrine cells that can produce gastrin or other hormones like gastrin from the small intestine as well. And like I said, I've got a list of these and we'll talk about these in just a minute, but let's work our way through this and see how this works because I've run out of room. Well, it depends on what you mean by hurts. There can be a lot of things. Obviously, overfilling the stomach can cause that distension of the stomach. If it stretches too much, that can cause pain. If there's too much acidity in the stomach, that can cause pain. Certain substrates, certain amino acids, or not amino acids, fatty acids, or things like that can affect the pH and can affect it. Things along those lines. And then there's also the cognitive aspect that can cause it as well. So again, there are several things that can cause stomach pain, right, or they have your stomach hurt. But those are the types of things typically. All right. So as we said, our cephalic phase can be either excitatory or inhibitory, right? You either like the food or you don't like the food. You're excited about the food or you're not excited about the food. So it's not usually both. Again, the key to the cephalic phase is it is before the food reaches the stomach, and it's typically initiated by some sense receptor in our head Again, as we talked about, this can be taste, this can be hearing, this can be smell, this can be sight, or like we also talked about, it can just be cognitive thought, just thinking about that food in expectation. And again, as we noticed, since it's primarily our excitatory, is primarily parasympathetic, is not primarily, is parasympathetic. As we talked about, that information primarily comes from the vagus nerves, as we talked about before. As we talked about, it increases saliva production, so that's what's most obvious to us. But again, we're focusing on stomach activity. And so it is going to increase the secretions by the stomach. And like I said, if it continues, it can also influence motility as well. All right, that is our cephalic phase. And then of course, remember, if it's inhibitory, then that is going to be sympathetic, and it will decrease motility. It'll decrease secretions. Gastric phase is primarily excitatory. Again, the stomach doesn't care what the food is or how it got there. It just wants to process what's inside of it. So again, this is primarily from a neural standpoint, our intrinsic or short reflex. We have some change in the stomach, a stretch, a change in pH, a change in osmolarity, or like I said, the presence of certain amino acids or certain fatty acids or things along those lines. And using that submucosal and myenteric plexus, using that short reflex, we are going to increase the rate at which the muscles contract, churning that food more and more. And we are going to stimulate the production of our gastric glands. But notice when we stimulate the production of this gastric glands, that causes the production of gastrin. And that gastrin also affects the smooth muscle, also affects the glands. So notice this gastric phase, remember we said, was both neural and hormonal. And as we said, depending on the composition of the food, in the volume of the food, it can take as much as 4 hours to empty the stomach. All right, so you have breakfast at 7, and by the time noon rolls around, you're ready for lunch. All right, again, I think those are pretty straightforward. Where things get a teeny bit trickier is when we get to the intestinal phase, because the intestinal phase has 2 parts to it. Right? We have made that bolus into chyme, and we are presenting that chyme to the small intestine. And in this case, our intestinal phase is going to be both excitatory and inhibitory. The excitatory phase is solely hormonal. Specifically, enteroendocrine cells of our small intestine produce intestinal gastrin. And that intestinal gastrin increases the activity of the stomach, increases the secretion of the stomach. But remember, as we talked about, this excitatory phase is a very brief effect. Because unlike the stomach that just cares that food is presented to it, the small intestine actually cares what the food is. And so based on the composition of that food, right? We want to modify the behavior of the stomach. And so this inhibitory component of the intestinal phase is both hormonal and neural. Notice we see the enterogastric reflex. This enterogastric reflex basically sends a signal to the stomach, decreasing the myenteric plexus. This decreases the motility. And again, if you're decreasing the motility, you're slowing the rate at which it presentation of chyme to the stomach. But this is a local effect. However, it also affects the long reflex as well. This enteroreflex will also, and I'll write it up here, it inhibits the vagus nerve, so we get less excitation, and it can actually excite the sympathetic pathway, which again, both of these things, inhibiting the vagus nerve and exciting the sympathetic pathway, decreases the activity of the stomach. So this enterogastric reflex affects both the short and the long reflexes. A strong neural component. However, we're also going to rely on hormones as well. We'll talk about the hormones in more depth when we get to the small intestine. But in general, as you can see here from the list here, there are intestinal hormones like gastric inhibitory peptidase, which as the name would indicate, inhibits the stomach, cholecystokinin, secretin, and all these types of hormones. In general, they tend to focus on either the motility and or secretion of the stomach. But again, the key to this one, and this is why we'll wait till we talk about it in the stomach and the small intestine. The key to this particular part is which hormones are released are determined by the composition of the food. Right. If the food is too acidic, we will want to release secretin, which stops the production of hydrochloric acid. And that would raise the acidity back up. Right. So this case, again, what's special about this hormonal inhibitory phase is that this is the first place really where the composition of the food matters. You could argue it matters in the cephalic phase, but if you think about it, the cephalic phase can be tricked, right? There's an amazing author who wrote a bunch of books about tricking your kids into hiding vegetables in their food, to which Jerry Seinfeld's wife then stole the idea and wrote her own book, and since she was more famous, sold more copies of hers. But you can hide broccoli in a brownie, right? You can make a tart where if you chop up the mushroom small enough, the kids aren't going to notice it's there. So you can trick the brain, but you can't trick the small intestine. If those acids are there, if those substrates are there, those hormones are going to be produced. So this is really the first place where the composition really matters as to controlling the activity, to make sure that the stomach is presenting the food at the right pace. All right. And so again, notice here we have all of that, the excitatory components, the inhibitory components, the cephalic, gastric, and intestinal phase, regulation of gastric activity. Questions on that? All right, excellent. So there you go. Our goal, and here we see the nice illustration showing this process. We have these big peristalic waves that are moving through our small intestine, our stomach. Again, remember I talked about that those pacemaker cells, the cells of Cajal, produce that basal rhythmic rate that can be enhanced neurally move through here, cause the massive churning. And notice when they reach the pyloric region, they cause a small amount to be expressed. This pyloric region holds about 30 milliliters of chyme. And each contraction, each of these contractile waves, these peristolic waves that go through, present about 3 milliliters of our chyme into the duodenum, into the beginning of our small intestine. So 3 milliliters at a time. So think about how long it takes to empty those 62 hot dogs out of your stomach into your small intestine when you're going 3 milliliters at a time. Is presented to the small intestine so that our small intestine is capable of processing that small amount of food with the maximal efficiency. All right, questions on that? Excellent. Love my stomach silences. That means you guys all completely understand what I did, which means I'm doing an amazing job. Or you're all asleep, one or the other. Excellent. Or I guess it's St. Patrick's Day. Y'all could be drunk too. All right, excellent. So on to the small intestine. Now, remember, one of the things we talked about, your alimentary canal in a cadaver from mouth to anus is 30 feet long. 20 of those feet is the small intestine. How the heck is something 20 feet long called the small intestine?
Diameters.
Exactly. The reason it is the small intestine is because it is small in diameter, not in length, small in diameter. It is by far the longest of all of our digestive organs. And anything 20 feet long can easily be divided into probably 200 different regions. And is that what we have in our small intestine? 200 different regions? Nope, just 3, absolutely, right? Those 3 are, and again, like most of the organs in the alimentary canal, they go from valve to valve. As we just mentioned, proximally, it is the pyloric sphincter between the stomach and the small intestine. And distally, it is the ileocecal valve between the small intestine and the large intestine. It is divided anatomically, but not evenly, into 3 distinct regions. The first, making up only about 5% of its length, only being about 10 inches long. And in fact, that's where it gets its name. Duodenum means 10 fingers. So it's only about 10 fingers long. The duodenum, duodenum, both are acceptable ways of pronouncing it. And more importantly, I don't care how you pronounce it as long as you spell it correctly on the exam. But the proximal part is the duodenum. The next part, the middle part, is the jejunum, making up almost 40%. And the remaining almost 60% is the ileum. In fact, that's where the ileocecal valve gets its name. The distal part of the small intestine is known as the ileum. The proximal part of the large intestine, this pouch-like region, is called the cecum. So not surprisingly, the ileocecal valve is between the ileum and the cecum. So that makes it easy. Now, notice we can clearly see where it connects to the small intestine. And we know that that is definitely the beginning of the small intestine, and that's the duodenum. We can clearly see where it connects to the large intestine. We clearly know that that part right there may be the ileum. However, what about this part right here? Is this part right here the jejunum, or is this one the ileum? Or this one back here, or this one here? Do we know for certain what is what? Is it perfectly segregated? All the jejunum on top, all the duodenum, all the ileum on the bottom?
No.
No. So while gross anatomy, it can be challenging just looking at this to tell what tube is what. When we look at this under the microscope, it is very easy to distinguish these 3 regions histologically. So on a microscope slide, will I show you a microscope slide of the small intestine? And not only do you need to be able to know that it is the small intestine, but you also need to be able to distinguish whether it is the duodenum, jejunum, or ileum? Absolutely. So you absolutely have to do that. So we'll make sure that we're capable of doing that. And I will tell you, it's super easy. There's one major trick, and we will see what that is in a bit. Like I said, the small intestine is the true workhorse of the digestive organs. It is indeed the major digestive organ. While we started the chemical breakdown of carbohydrates in the mouth and proteins in the stomach and lipids in the stomach, the completion of the mechanical and chemical digestion occurs in the small intestine. And I should point out because we can't forget our last macromolecule, all of the chemical and mechanical breakdown of our nucleic acids. Oops. Still can't spell it right. There we go. Ugh, good gravy. Occurs in the small intestine. The small intestine is the primary organ of absorption. We talked about how there's a little absorption that takes place in the oral cavity. We talked about a little absorption in the stomach. But the majority of both our substances and our water occur in the small intestine. I know some people think that the large intestine is the primary organ for water absorption, and that is not correct. The majority of the water that is absorbed by the digestive system occurs in the small intestine. We'll talk about why that inaccuracy is often believed later when we talk about the large intestine, but for now, we'll talk about this. As we know, all of the intestinal blood vessels don't travel straight back to the heart. Where do they go instead? Where do all of the intestinal blood vessels go? There we go, to the liver, absolutely. Via what?
The hepatic portal.
Hepatic portal vein, absolutely. Excellent, excellent, excellent. And most of this digestive function occurs in the proximal portion. As I said, the small intestine in general is highly specialized for its job, but the three regions are also histologically very distinct from each other, and we will easily be able to tell them apart. Let's talk about the general specializations that we see here, starting with something similar to what we saw in our stomach. Notice if we look closely at this illustration, we can see that there is a big fold that is comprised of both the mucosa and the submucosa, forming a big, huge fold, very similar to a rugae. Of course, these aren't called rugae. What are they called?
Pica.
Say again? Sure. Okay. The plicae circularis, which absolutely is an appropriate term for it. However, another appropriate anatomical term for it are the circular folds. So I will allow you to use either of those terms. Now, rugae involved the mucosa and the submucosa, and they allowed for expansion. The plicae circularis also also involve the mucosa and the submucosa, but what is their function? Does our small intestine expand like our stomach does to accommodate 62 cheeseburgers or hot
Does it
dogs?
increase the surface area?
Exactly. Absolutely. They're there to increase the surface area. Notice if we look closely at just the mucosa, and in this case, notice just the mucosa. This is actually mislabeled. So let's do this. This is not the submucosa. This would be the lamina propria. With that lamina propria, we would then see the muscularis mucosa down here like this. So just involving the mucosa. Kind of like the gastric glands. However, remember gastric glands were folds into the mucosa. In this case, we have finger-like extensions of the mucosa that stick out. And what do we call these finger-like extensions of the mucosa that stick out?
Villi.
Villi, absolutely. And what is their function?
Increase surface area.
Increase surface area. And lastly, look at the epithelial cells that line the villi. These epithelial cells are known as absorptive cells. They are, of course, simple columnar, because again, all of the small intestine is lined by simple columnar cells. But notice they have finger-like extensions of their plasma membrane that extend out on their apical surface. And what do we call those special organelles?
Microvilli.
Microvilli. And what are the functions of these microvilli?
Increase surface area.
There you go. Notice there are these three major specializations to our small intestine. The mucosa and submucosa form big folds, and these big folds are the plicae circularis, the circular folds. On those circular folds, lining the entire surface of the circular folds, are small villi sticking out with those finger-like extensions to increase surface area. And lining the entire surface are epithelial tissue, the simple columnar epithelial tissue, contain microvilli, which increase surface area. We have these three massive specializations that help to increase surface area. And of course, why do we want this big increased surface area?
Increase absorption of nutrients.
Yeah. Absorption, absolutely. In fact, notice if you look closely at the villi, we'll see some things that we are familiar with. Are capillaries for the absorption of nutrients into the blood, and also those specialized lymphatic capillaries called the lacteals for the absorption of lipid-soluble materials via digestion. So we have these massive, massive specializations, and these massive specializations dramatically increase the surface area. Over as much as 600 times the surface area of if it was just a hollow tube. It is a massive, massive increase of surface area because, again, this is where the massive influx of substances are going to occur. Obviously, the plicae circularis also play an important role in slowing the chyme, making more contact with it, so that, again, we can absorb the materials into our capillaries, into our lacteal, to send it to the liver and send it to our blood and send it to our heart and send it on its merry way. Now, our small intestine does not have the protection in it that our stomach does. So the chyme coming in from the stomach, the digestive enzymes that are going to play a role can be very aggressive with the lining of our small intestine. We are constantly shedding these epithelial cells. In fact, an epithelial cell in your small intestine typically only lasts about 3 days. So every 3 days, you have a completely new epithelium to your small intestine. You might not have thought of it in those terms, but maybe you know someone who has had to deal with chemotherapy, right? Dealing with some kind of cancer. One of the things that commonly happens, chemotherapy is basically putting a toxin in your body that targets rapidly dividing cells, because that's what cancer is, uncontrollably rapidly dividing cells. However, it also targets other cells that divide rapidly, like the matrices of your hairs. So hair often falls off if you take chemo. People who take chemo often have major digestive issues because, unfortunately, that chemo also targets these rapidly dividing epithelial cells of our small intestine. So it's quite common to have intestinal discomfort when someone is dealing with chemotherapy. All right. Questions on that? All right, those are the specializations. Here we see the pretty picture. And remember one more thing we talked about with the small intestine. The duodenum of the small intestine, remember, is retroperitoneal. So it is covered with an adventitia and is located behind the parietal peritoneum, outside of the parietal cavity, whereas the jejunum and the ileum are both intraperitoneal, which means they are both surrounded by a serosa in the abdominal pelvic cavity and are also anchored to the posterior wall by, remember, what we call the mesentery proper, which is the specific name of the mesentery associated with the small intestine. Obviously, from a gross anatomy standpoint, that's an easy way to distinguish them, but we want to do it histologically as well. And here's some more pictures. Again, doing a nice job. Notice, again, these illustrations do a great job of showing both the circular folds, which involve both the mucosa and the submucosa, and the villi, which just involve the mucosa. But remember, on top of those, lining those villi, are those absorptive cells with the microvilli. So we can't forget that. And notice this must be either the ileum or the jejunum, because notice it's surrounded by a serosa, which comes together to form our mesentery, containing the blood vessels and nerves that carry information and blood to and from our intraperitoneal organs, small intestine. More pretty pictures showing this as well. Again, notice this is just showing the villi. We can tell that because there's our muscularis mucosa. So this just involves our mucosa. So these are indeed the villi. Again, here we see a plicae circularis with villi on top of it. Again, your book's got lots of pictures that show that. And lastly, here we have our first histology view of this. This particular histology view shows us a couple things. Notice at first when you look up here at the mucosa, and again, we can tell this is the mucosa because one of the first things you should look for is the muscularis mucosa. Once you find that muscularis mucosa, you know everything above that is the mucosa. And the first thought may be that this could be the stomach, right? These kind of look a little like gastric pits, and that's because these are actually gastric glands. Pardon me, not gastric glands. These are intestinal glands. These intestinal glands primarily are producing both mucus or mucin and bicarbonate. What's bicarbonate? Anyone know what bicarbonate is? My microphone's on, right? Come on, someone's got to know what bicarbonate is. Oh, wait.
A salt containing HCO3.
True, although rather than a salt, I would think of it as a buffer. Right, it is a buffer. You've got the right idea. It is HCO3 negative. It is an ion, but its primary job is to act as a buffer to help to neutralize acidity. Remember, that chyme coming in from the stomach is at a pH of 1 or 2. Do we want to be rubbing a pH 1 or 2 chyme on the naked surface of our small intestine? No, we want to help to neutralize that. So we can neutralize that with that bicarbonate. It still kind of does look a little bit like stomach. However, there's a couple key features we can notice. The first is, notice we have the villi extending out. We don't have villi in the stomach. So that's something that can help us to distinguish that as well. However, the easiest way to tell this is not the stomach is to come here to the muscularis. And when I come here to the muscularis, I see one very clear layer. I see a second very clear layer. Do I see a third very clear layer here in the muscularis? Nope, I only see 2 layers. So that is absolutely positively, very clearly not 3 layers. So that is definitely not the stomach. So that tells me right away that it is the small intestine. So then the next big question becomes, how do I tell what part of the small intestine that it is? The easiest way to tell what part of the small intestine you are looking at is to look in the submucosa. When you look in this particular submucosa, what you see are a massive amount of glands. These glands are helping to produce extra mucin and extra bicarbonate, and their job is to help to neutralize that chyme when it first enters into the small intestine. Now, these glands were first discovered, of course, by good old Bob Brunner. So they are called Brunner's glands. However, the other name for these glands are duodenal. There we go. Duodenal glands. And guess what part of the small intestine duodenal glands are located in?
Duodenum.
The duodenum, there you go. So it's perfectly acceptable to call them Brunner's glands. Brunner was the one who first discovered them, but they're also known as duodenal glands, and that helps because that tells us we're in the duodenum. If you see these Brunner's glands, if you see these duodenal glands in the submucosa, it is a dead giveaway you are looking at the duodenum of the small intestine. Notice the first place I told you to look was in the submucosa. Now, again, if you didn't do that, one of the things you might have noticed is once again we have villi. Notice these villi seem to be a little bit more extensive in their length. We still have some intestinal glands here, but I told you to look in the submucosa. And when you look in the submucosa, what do you see in this submucosa? What the heck are those things? Follicles, malts, but not just any malts. We have a large collection of lymphatic follicles all clustered together in a portion of the small intestine. And didn't we have a fancy name for those? Peyer's patches. And if we see Peyer's patches, what part of the small intestine are we in?
The ileum?
Ileum. There you go. See duodenal glands? You're in the duodenum. See Peyer's patches? You're in the ileum. And what do you see here? I see a muscularis. I see a mucosa with villi, pretty pronounced villi. Some mucus glands, some some intestinal glands. But again, when I look in the submucosa, do I see Brunner's glands? Do I see Peyer's patches? No. So if I see neither, what part of the small intestine am I in?
Jejunum.
There you go. If you don't see Brunner's glands, you're not in the duodenum. If you don't see Peyer's patches, you're not in the ileum. So if you're not in the ileum and you're not in the duodenum, you must be in the jejunum. So like I said, it can be a little scary, but once you see those villi, once you figure out you're in the small intestine, then all you have to do is look in the submucosa and it will tell you precisely what region of the small intestine you are in. You either have Brunner's glands, have Peyer's patches, or have neither. And it really is just that simple.
Can you flip through those one more time?
Nope.
Please.
There you go. Yeah, notice that I'll do it quickly. Notice all three of them have villi. Notice all three of these are different magnifications too, right? So again, doesn't matter the magnification, doesn't matter anything else. Find the submucosa. If you find these simple tubular glands that look very clear because they're producing a lot of mucus, a lot of bicarbonate, those are those Brunner's glands. Big, prominent glands. Notice we go back to this one. You can see a blood vessel here. This may be a duct of a gland or a portion of a gland or something like that. But here, massive collections of them. This is clearly, obviously, big, massive glands in the submucosa. Duodenum. This is clearly a big, massive collection of Peyer's patches in the submucosa. Clearly dealing. And here we have neither. We don't have any malts. We don't have any major glands. We have neither. Now notice there are other differences. The ileum, the jejunum typically has the largest villi. Notice the duodenum tends to have shorter ones. The ileum tends to have shorter ones. These tend to be the longest and largest, but that's very subjective. And especially if you're just looking at one of these, because magnification can change and other factors can change. It's very, very subjective. What's not subjective is what's going on in the submucosa. So look at the submucosa and it will clearly tell you what region of the small intestine you are in. All right. Questions on that? All right, excellent. This is a good point for our second break. I think this is a good point for our second break. Where are we time-wise? Let me see what I have left to do today. Oh yeah, this will work. Okay, perfect. Okay. So this is a good point for our second break. Let's go ahead and take our second break. It is 10:35, so that means we will come back at 10:50. And I will start the recording at that point. All right, so any questions before we take our second break? All righty, I will see you guys in 15 minutes. All righty, we have finished our anatomy of the small intestine, both gross and microscopic. So now we get to focus on is the intestinal activity. Again, intestinal activity includes the beginning of the intestinal phase when we're controlling our gastric activity. Again, this happens when the chyme enters into the duodenum. And remember, as we talked about, this is going to be where we're going to start to be able to modify behavior based on the composition of the material that is in that chyme. In particular, some of that involves stretch receptors. When our chyme enters into the duodenum, those stretch receptors stimulate the production of intestinal juice. And again, depending on the composition of the food, the amount of times you eat during the course of the day, the volume of the two foods that you eat, it can vary dramatically, but on average, we produce about 2 liters of intestinal juice during the course of the day. This is mostly water, some mucin, and as we talked about, bicarbonate ions. Those bicarbonate ions are going to neutralize the acidity. Again, this is an important chemical buffer. that actually comes from, and we'll talk about this a lot when we get to the respiratory system. But basically what happens is when carbon dioxide mixes with water, the result of that is that it becomes carbonic acid. However, carbonic acid, like all acids, are hydrogen donators. So what happens is the hydrogen ion disassociates, and what you're left with is HCO3, which is a negative ion, and that is the buffer. And what's great about this process is it is a reversible chemical reaction. So what can happen, this isn't chemistry, this is biology. What can happen is as there is an increase in the amount of hydrogen ions that are present, that bicarbonate can mix with it and start neutralizing that acidity, producing water and carbonic acid, which again leads to some of the gas in our digestive tract and can be absorbed and passed out through our respiratory system as well. So this is an important chemical buffer that plays an important role in helping to neutralize the acidity. Lastly, also, as we mentioned, our intestinal juice will include those hormones, but again, those hormones are going to be specific to the composition of it. Now, regulation of intestinal activity does start as early as the presentation thinking, hearing, smelling, seeing food. Again, we talked about how it affects the stomach, but we also mentioned it is going to affect other organ systems as well. In particular, primarily the influence it's going to have on our small intestine is to get the small intestine ready for new food, right? It is basically processing chyme. But seeing food, smelling food, tasting food, food hitting our stomach, all of those things tell the small intestine, hey, more food is on the way. You need to be finished processing what you've already got inside of you and make room for more. So we are going to get long reflexes that are going to influence this, starting from when we start thinking about or smelling or seeing or tasting food. Local reflexes, so again, notice this is both our intrinsic and extrinsic controls. And again, we're going to have those hormones that are being produced as well. I keep talking about how these hormones are produced based on the composition of the chyme. Let's talk a little bit about how this works. For instance, as stretch receptors expand to accommodate the food that comes in, that neural reflex, as we talked about, causes the production of mucus from the goblet cells, activity of those Brunner glands found in the duodenum, because again, our goal here is to neutralize the acidity. We've got that very, very acidic 1 to 2 pH chyme entering into the small intestine. And if the small intestine is going to absorb nutrients, we can't have it lined with a thick, protective, viscous, alkaline mucus. It needs to be exposed to the food. And so we need to neutralize that acidity. Now, if the composition of the food contains a fair amount of proteins, remember we said proteins are relatively easy to be able to break down. So proteins lead to the production of gastrin. Gastrin, as we know, is going to be excitatory to the stomach, causing it to increase in its activity, but it is also excitatory to the small intestine. where it increases the motility of the small intestine. In fact, if you remember, we talked about that process where the circular layers will go in that alternating fashion back and forth. They actually help to stimulate that process we called segmentation. And if you don't remember it, don't worry about it. We'll talk about it again in just a minute. But gastrin does more than that as well. Remember, we talked earlier about how kids sit down, have a bite or two of food, and then have to jump up and run to the bathroom. Because remember, we said gastrin influences other organs as well. And one of the things gastrin does is it opens the ileocecal valve. Remember, our goal here is not just to get the small intestine ready for new food, but we got to get rid of the old food as well. So by opening the ileocecal valve, that helps to release the old food into the large intestine to make room for new food. And if you're a little kid who has a very small large intestine, that presentation of chyme to the large intestine can often be enough to trigger our defecation reflex, which we'll talk about later. So again, our goal here is to get rid of the old chyme, to the large intestine to make room for new. However, remember we talked about how lipids and some amino acids, but primarily lipids, are harder to break down. When lipids are present in our small intestine produces a hormone called cholecystokinin. The bad news is you have to spell cholecystokinin. The good news is once you spell it once, you can abbreviate it CCK, because that is the abbreviation for cholecystokinin. So once you've written it out once, you can abbreviate it CCK. And as we talked about, cholecystokinin helps to process those more hard to digest lipids. And there are a couple really important ways that it is going to do this. One of the major targets is, of course, the stomach. If the meal we have ingested has a lot of lipids in it, we want to slow the rate that it is sending the food to our small intestine so we have more time to process it. So to the stomach, this cholecystokinin is going to cause a decrease in motility. So the food is presented more slowly. Right? But we need additional help to break down those lipids. And someone earlier mentioned bile. What makes bile?
Liver.
Liver. So cholecystokinin is going to stimulate the liver to make and release bile. Now, your liver is making bile constantly. Does that mean you're constantly releasing that bile into your small intestine?
No.
No, when you're not releasing it into your small intestine, where are you sending it?
Gallbladder.
Gallbladder. And what's special about the gallbladder is not only does it store the bile, but it concentrates it. While that bile sits there inside of the gallbladder, it draws water out of it, making it much more concentrated. In fact, the bile in your gallbladder can be as much as 10 times as potent as the bile in your liver. So again, this cholecystokinin is going to stimulate the gallbladder to release its concentrated bile.
So is there any issues when somebody is missing their gallbladder?
I'm sorry, I didn't quite hear that. What?
So is there any issues when somebody is missing their gallbladder?
So great question. So when someone's gallbladder is removed for gallstones or something along those lines, it can be an issue it's going to limit your ability to be able to properly digest fats. Now notice the gallbladder doesn't make the bile. The liver is what makes the bile. So you're not going to be without bile. And in fact, the ducts of the liver will expand a little bit so that it's able to store a little bit more of the bile. But what the liver can't do is the liver can't concentrate it the same way the gallbladder did. So the bile that is released from this cholecystokinin won't be as concentrated, and so won't be quite as effective. Right. So if you were eating three scoop ice cream sundaes before you got your gallbladder removed, and you're still trying to eat 3 scoop ice cream sundaes, that may be a little much for your digestive system to be able to fully digest. And if you're not able to fully completely break down those fats, you can get intestinal cramping, discomfort, diarrhea, and stuff like that as a result of it. So you may have to switch to a single scoop ice cream sundae instead of 3. Right. So it limits how effective you are breaking down bile, but it doesn't, breaking down fats, but it doesn't eliminate your ability to make and store bile. It just decreases it and decreases its efficiency. Great question. All right, excellent. Now, these things are released along with pancreatic. So there's going to be some pancreatic activity where we're going to produce pancreatic enzymes. And it turns out both the pancreas and the liver and gallbladder all use the same duct to release their materials into the small intestine. And so there is a single valve that basically controls the release of both of those components. So not surprisingly is an alphabet soup name that tells you exactly what it is. It is the pancreatic sphincter. And this hepatopancreatic sphincter is relaxed by cholecystokinin so that we can release the bile, we can release the pancreatic enzymes into the small intestine, and more specifically into the duodenum. So as you can see here, the difference between lipids and protein is dramatically different. This hormone, cholecystokinin, has a lot more important effects and influences because it's going to take us, it's going to be harder for us to break down these lipids. So I've listed it all out there. I think I have an abbreviation of all that stuff here. Again, pancreatic enzyme stimulates the liver, contraction of the gallbladder, relaxation of that hepatopancreatic sphincter. So we got all of that on there. Excellent. But we also explained why and why those are important. And what I don't have here, it looks like, is remember that it also inhibits the activity of the stomach, or more specifically, the motility. So let's not forget that as well. Excellent. All right, so enzymes from the pancreas, bile from the liver, concentrated bile from the gallbladder, relaxing that hepatopancreatic sphincter, and inhibiting the motility of the stomach, all because of the presence of lipids. Let's talk about another example. If there are a lot of carbohydrates, certain fatty acids, but primary carbohydrates, we produce a hormone called gastric inhibitory peptide. As the name would indicate, its primary its primary target is the stomach, where it is going to inhibit the secretions of the stomach. Why might we want to inhibit the secretions of the stomach if the food is very carbohydrate based? Isn't that pepsin, isn't that hydrochloric acid really, really important for the digesting of foods?
No, so you don't want the pH to drop very low?
Yeah, it is. Hydrochloric acid and pepsin are very, very important for the breakdown of proteins. But notice our meal isn't very protein rich. So it doesn't help us to make the chyme really acidic if all we're having to worry about is the breaking down of carbohydrates. Absolutely. So we don't have to neutralize that acidity. So we still bring it on motility wise, keep bringing me that food. I can break down glucose, but I don't need the hydrochloric acid. I don't need the pepsin because again, I'm not dealing with proteins. However, I am dealing with sugars. And if there's a lot of sugars in my alimentary canal, that means there's going to be a lot of sugars in my blood. And so one thing that the gastric inhibitory peptide can do is actually stimulate insulin release. What releases insulin again?
Pancreas.
You get partial credit for that, but we can be more specific after the last section.
B cells.
Beta cells, absolutely. Those beta cells in the pancreas release that insulin. Because again, if there's a lot of sugar in our alimentary canal, there's about to be a lot of sugar in our blood, and we need that insulin to be able to help to store that glucose in the muscles, in the liver, right, in the adipose to maintain appropriate blood glucose levels. If the chyme is too acidic, our small intestine can release a hormone called secretin. And as its name indicates, it inhibits the secretion of hydrochloric acid from the stomach, trying to bring the pH of the chyme back up. And we want to slow the motility of the stomach. We want to slow the presentation of this really acidic chyme into our small intestine. So we have time to produce the bicarbonate, time to produce the mucus, time to produce the water, so that we can help to neutralize that acidity. And not surprisingly, secretin also stimulates the pancreas, in this case, not to produce more enzymes, but instead to produce more bicarbonate. Again, with the goal there of being able to neutralize that acidity, bring the pH back up so that our small intestine isn't damaged. Right? We keep talking about this protection of the small intestine from that chyme, but what we didn't talk about is the opposite. What happens if you have a very weak, lower esophageal sphincter, right? And especially if you're producing a lot of acid in your stomach, you can get that acid reflux, where the chyme starts to move back up into the esophagus. Right? Why is that a problem?
It's disgusting.
Okay. It isn't necessarily right.
Painful.
It isn't necessarily an enjoyable experience or taste necessarily associated with that, but more importantly, it can be damaging to the wall of the esophagus. The esophagus doesn't have the mucus to protect it the same way the stomach does. It doesn't have the ability to make bicarbonate the same way that the small intestine does. It doesn't have the hormones to tell the stomach to stop making the acid. So chronic acid reflux can cause major damage and major ulcers to that esophagus, right? So we don't want that happening in the small intestine. So the small intestine has protection because that's the way the chyme is supposed to go. The chyme isn't supposed to come back up. And so that's why that acid reflux can be so damaging to the esophagus, because it doesn't have the protections, because the food isn't supposed to move that direction. Your book has a nice list of more of the hormones. Histamine, as we talk about, has some effect. There's some other ones in here we didn't talk about, motilin, somatostatin, things along those lines. Focus on the ones we've talked about. But for the ones we've talked about, like cholecystokinin, know the targets, know the function, know the stimulus for their production. So those that I've listed, make sure you understand what it is that they do. All right. And of course, your book's got one of these pretty flow charts that show this whole process as well. Again, take home for all of these neural and hormonal controls is we want to control the rate at which the stomach is presenting stuff to the small intestine. so that the small intestine has the time to both chemically and mechanically digest the food. As I mentioned, our intestines have some different types of motility than what we see in the stomach and the esophagus. Of course, they are going to undergo peristalsis the same way for the propulsion that we talked about before, but there are other specialized movements of the small intestine. The primary one that we talked about earlier is that process of segmentation. Remember, segmentation is where we get an alternating contraction of portions of the circular layer of the muscularis, right? Remember, peristalsis, we're alternating between circular and longitudinal, circular and longitudinal, circular and longitudinal. Now, in this case, circular over here, then circular over here. And then in this alternating fashion, it is moving the chyme back and forth within the small intestine. Notice with this, there is very minimal forward movement. Right? Instead, the primary function of this is twofold. First is mechanical digestion. It is going to continue to mechanically break down the food. Basically, this increases the surface area, making it easier for the enzymes to interact with the food. And the other thing that it is doing is it is basically rubbing the food across the surface of the small intestine. This is important for two reasons. Well, you could argue even three. One, it slows it down. So again, longer in the small intestine, more time to process what is there. But rubbing it back and forth, right? Remember, we're going to talk about in a minute all of the enzymes that are being released by the pancreas. The pancreas is one of the primary organs whose exocrine function, those acini of the pancreas, release a massive amount of enzymes to help in the breakdown of our foods. But on the surface of the absorptive cells, right? Remember, we have these absorptive cells, simple columnar cells with the microvilli, the non-motile microvilli on top of them. On these microvilli are a bunch of specific enzymes that help in the breakdown. There are literally dozens of these different types, and collectively, they are called the brush border enzymes. I will not hold you responsible for any of the names of the specific brush border enzymes. You just need to know them collectively as brush border enzymes and know that they play a role in the breakdown of the, let's be more specific, of the chemical breakdown of all macromolecules. Carbohydrates, proteins, lipids, nucleic acids. There are brush border enzymes that help and assist in the process of all of that. So if we're rubbing it back and forth across the surface of these absorptive cells, we are increasing their interaction with these brush border enzymes. And again, we are increasing the chemical breakdown. Of course, the other advantage of rubbing them back and forth across these microvilli is that essentially increases the surface area, or you can think of the contact with the cells for more absorption to take place. So we get that absorption that is going to take place there from rubbing that food back and forth across the surface. So the segmentation doesn't get food from point A to point B, but by mashing it back and forth, we continue to break it down. We continue to mix it with enzymes, rub it against the surface of the cells to help to both mechanically break down, chemically break down, and also to assist in absorption as well. We still have to move things through it, and so we're still going to use peristalsis for doing that. That definitely occurs in the small intestine as well. And these processes of segmentation peristalsis can be, not surprisingly, controlled both hormonally, but also neurally. A couple of the important reflexes, one of them is what we call the gastroileal reflex. When you have that cheeseburger, it stretches the stomach, and that again increases motility and secretions in the small intestine, because again, we need to get rid of the old to make room for new. So we have that gastroileal reflex that helps in that. Again, relaxes that ileocecal valve. Remember, gastrin releases that so we can get it out of the ileum into the large intestine to make room. And again, depending on the volume, depending on the composition, the frequency of eating, it can take anywhere between about 3 and 6 hours for the small intestine to empty itself completely. All right, questions on that?
How is fasting beneficial to all of this? So if you don't have food and stuff going into your stomach and intestines, say for like 12 hours or 24 hours even.
I'm not sure what you mean by saying that it is helpful.
Well, I guess it's generally known as a good thing.
I believe he's talking about intermittent fasting.
So yeah, so with some of these processes like intermittent fasting, the goal of things like that isn't necessarily that it is helping the process of your digestion. The advantage of going a longer time without eating does give more time for more thorough processing of the food, so it may be a little bit more efficient that way. But most processes like fasting or intermittent fasting are either done more for religious reasons, or they are done for general health reasons. The intermittent fasting seems to, and if done properly in the right ways, has the potential to lead to a general increase in metabolism, and like the keto diet we were talking about earlier, it can help to encourage the body to be more likely to break down lipids as opposed to carbohydrates for the production of energy. So again, it can be useful for things like losing weight or increasing overall general metabolism that way. I don't think it necessarily directly affects the digestive system. Like I said, I think the one thing it would do to the digestive system is if you're not constantly shoving food in your mouth, your small intestine does have more time to process the food that's in it. So it might be slightly more efficient because things will go through it a little bit more slowly. And so emptying that way. So I guess it could be potentially beneficial for the digestive system that way. But I think, like I said, those things are more done for either cultural, religious, or typically weight loss related regions, which don't necessarily benefit the digestive system directly. It isn't necessarily harmful, right? Obviously, starving yourself, which is essentially what fasting is, if not done properly, can cause implications and affect metabolism and things along those lines. But when handled properly, under the guidance of a doctor or nutritionist or something like that, it can be an effective way, or it appears to be an effective way, that people have successfully lost weight. All right, great question. Any others? All right, We've talked about the small intestine. I want to talk more about these three organs that we just finished talking about. How are we on time? We're doing good. That are associated with the small intestine, and primarily, as you can see here nicely with the illustration, are associated with the duodenum. And they are, again, the liver, the gallbladder, and the pancreas. Notice as we look carefully at this illustration that puts them all together, we can see a couple things. Notice how there is this main pancreatic duct that goes down the center of the pancreas. Again, remember the pancreas is a mixed gland, and that is what we see here. As we learned in the last section we focused, red's not going to show up well, on here, the pancreatic islet, which has the endocrine function, which again releases insulin and glucagon and other hormones into the blood. But we also talked about how we have out here all these ball-shaped pancreatic acini. And they're the ones that are producing the enzymes. They're the ones that are producing the bicarbonate and are releasing it via a duct, the primary pancreatic duct, into the duodenum. Notice also, we can see that both the liver, which produces bile, and the gallbladder, which stores and concentrates bile, also have a common bile duct that come down and attach to that main pancreatic duct. And both of them come together into a structure I will point out to you later, the better picture, to a finger-like extension that sticks out into the duodenum known as the duodenal papillae. This duodenal papillae has a big, huge ampulla inside of it, known as the hepatopancreatic ampulla, where both the secretions of the pancreas and the secretions of bile from the liver and gallbladder can be released. And as we talked about, this here in the enlargement of the circular layer of smooth muscle is where we have that hepatopancreatic sphincter. And I'll show you a better picture of that in a moment. However, there is one other thing that I want to show you. Notice the pancreas does have an accessory duct that also by itself can release substances into the duodenum. So it is possible for it to release some of its secretions without having to release bile through it as well. Here we get a little bit of a closer up look, but I think there's the picture I wanted. Notice here we have that common bile duct coming down from the gallbladder and from the liver. That main pancreatic duct coming in here. Notice the enlargement of the circular layer forms a valve known as the hepatopancreatic sphincter, leading into this big, huge hollow space known as the duodenal or hepatopancreatic ampulla, which extends out into that finger-like extension we saw on the illustration, the duodenal papillae, releasing these secretions out. Here's a nice illustration of our pancreas, reminding us that this is a mixed gland. Here we have the islets with its blood vessels, so it can make its hormones and release them into the blood. Whereas here we have our ball-like acini producing the bicarbonate, producing the enzymes and releasing them into those ducts, which are then going to release it outside the body into the small intestine. Again, hopefully I don't have to do too much with the anatomy of the pancreas because we looked at that already in the last section. But like I said, we focused on, or histologically I mean, because we did that in the last section. But from an exocrine function standpoint, again, depending on the composition of the food, the number of times you eat during the course of the day, the volume of the food you eat, it can vary dramatically. But on average, an individual produces about 1.5 liters of pancreatic juice during the course of the day. Obviously, this is going to contain water, ions, and a massive amount of bicarbonate and other phosphate buffers. Pancreatic juice is one of the more alkaline secretions that we make with a pH about 7.5 to 8.8, which again makes sense. This is being released right into the duodenum, right where that really acidic chyme is coming in, and we want to neutralize it, protect the small intestine. And so this bicarbonate and these phosphate buffers go a long way towards protecting that small intestine. Of course, this is going to be controlled by the sympathetic and parasympathetic nervous system that can start as early as when we think about food or taste food or smell food. But remember, this is also going to be affected by hormones like cholecystokinin, which triggers the release of enzymes, and also secretin, which I should have here. Oh, I don't have it. Also secretin, remember we talked about secretin, which causes the release of more of the bicarbonate. So cholecystokinin releases the enzymes, secretin releases the bicarbonate. So our pancreas is controlled both neurally and hormonally. And if you think of the islets, those islets, remember, are controlled humorally by glucose levels. So all three of our types of control of glands we can see here in our pancreas. We've talked about the importance of the bicarbonate and the other buffers. But as I mentioned, the pancreas is the primary organ that is providing enzymes to break down all of our macromolecules. For instance, our pancreas produces primarily trypsin, but other hormones like chymotrypsin and carboxypeptidase. Any idea what these enzymes break down? Don't worry, it gets easier after this one. Well, you have 4 macromolecules, so guess. You have a 25% chance of being right.
Peptides.
Right, proteins, absolutely. This is going to break down proteins, absolutely. All right, hence the peptidase breaks down that. Pancreatic amylase, guess what that breaks down?
Starch.
Carbohydrates, absolutely, excellent, starches. Pancreatic lipase, guess what that breaks down? Again, these should be getting easier.
Fats.
Yeah, fats, lipids, absolutely. Ribonuclease and deoxyribonuclease, guess what those break down?
The DNA.
Yeah, and RNA, right? This breaks down nucleic acids, absolutely. So again, our pancreas produces enzymes that are capable of completing or helping to complete the breakdown of all of our macromolecules. All right, so whatever it is that you ingest, right, we have the keys here to break those bonds for absorption. Now, remember how we talked about pepsinogen is released from the chief cells in that inactive form and is then activated into the active pepsin to protect the cell that produces it. So not surprisingly, much of the same occurs in the pancreas. Most of the pancreatic enzymes are actually released in an inactive state. And typically they get activated by those brush border enzymes in the small intestine. Now, your book actually does a really nice job of showing one of these pathways. I'm not going to hold you responsible for this particular these pathways, but I just want you to see how this works. So for instance, our pancreas releases trypsin in its inactive form, trypsinogen. Trypsinogen, when it reaches the small intestine through that duodenal papillae, is activated by one of those brush border enzymes that is located in the small intestine, right? And then with that enteropeptidase, and again, you don't need to know these words, and it is activated into trypsin. Trypsin, like pepsin, can help to activate more trypsin and can help to activate chymotrypsinogen into chymotrypsin and pro-carboxypeptidase into carboxypeptidase. But again, you don't have to know the inactive and the active form. I like this picture because it does illustrate and show that, like we saw in the stomach, most of these are going to be expressed in an inactive form and will be activated in the small intestine to protect the cells of the pancreas. So again, you're not responsible for these specific pathways or these specific names, or at least not the inactive names. You need to know these and what they do. And remember, all of these do proteins, but you don't have to worry about the pathway associated with activating them. Just know that it happens. All right, so there you go. That is the pancreas. Questions on that? Are we on time? All right, what that leaves us with is the liver and the gallbladder. This is going to take a little bit more work. So I want to start first by talking about the gross anatomy of the liver. But I think we're going to save, let me see where we are time-wise. Hold on one second. Say Tuesday or Thursday. I don't remember. Or Monday or Wednesday. Well, today's Wednesday, right?
Wednesday.
Today's Wednesday. Okay. All right. So let's see how far we can get. Let's talk about this because I do want to. We're going into the weekend, so let's cover. I want to cover some of this so you guys have some time to think about this. All right. So let's see how far we get. All righty. Our goal is to talk now about the liver and gallbladder. Here we see the liver. Here we see the head of the gallbladder peeking out here from the bottom of this. The liver is your largest visceral organ, the largest organ of your abdominal pelvic cavity, taking up most of the right upper quadrant. And you can see it is very asymmetrical. It is actually comprised, and though you can't see it fully from here, of 4 distinct lobes. This here we see an anterior view. And as you look in this anterior view, you can see the very large right lobe of the liver and the much, much smaller left lobe. These lobes can clearly be distinguished from each other by a large connective tissue fascia that separates them, known as the falciform ligament. So this falciform ligament clearly differentiates the right lobe from the left lobe of our liver. Interestingly, inferior, where that falciform ligament begins, there is what appears to be an extension of connective tissue known as the ligamentum teres, or the round ligament. Anybody know what this round ligament happens to be? No? Well, related to bile is definitely a good guess, but as it turns out, that's not the case. This is actually a vestigial structure from when you were inside mommy's tummy, right? Because when you were inside mommy's tummy, and let's draw the wall of your tummy there and the wall of mommy's belly there, right? Mom's belly had that nice big placenta sticking on it. And off of that placenta was an umbilical cord that connected to you. Well, that umbilical cord's job was to bring nutrients from mom, and guess where it goes? Straight to the liver. So we have these umbilical veins that come from mom back to the liver. The same way our digestive system goes to the liver first, our placenta went to the liver first. Of course, when you're born, we cut that umbilical cord. It shrivels up and becomes your belly button, but it shrivels up on the inside as well. That umbilical vein shrivels up and becomes this vestigial structure known as the round ligament, or ligamentum teres. So it's basically the leftover dried up umbilical vein from when you were being fed by mommy. When we look at the posterior side of the liver, one of the interesting things we see about this is this nice big indentation it basically posteriorly wraps around. Sorry, I'm going to go ahead and do that. It wraps around the inferior vena cava. So we can see this nice big indentation in the liver where it wraps around the inferior vena cava. And from that, we can see the much smaller posterior lobes. And those smaller posterior lobes are the superior caudate lobe and the inferior quadrate lobe. So these two lobes are adjacent, right next to the inferior vena cava that travels by, and they're in alphabetical order. Caudate comes before quadrate. Caudate is superior, quadrate is inferior. Right? And so here we see that posterior view. And of course, this over here is that smaller left lobe, and this over here is the larger right lobe. So we have this nice, very simple, basic, gross anatomy to our liver. And the liver itself, histologically, is divided into somewhere around 500,000 hexagonal liver lobules. These liver lobules are both the functional and structural units of the liver. They're formed by cells known as hepatocytes. So let's actually take a look at this. Let's actually draw some of this. So, as I mentioned, we have these connective tissue septums that divide the liver into about 500,000 hexagonal shaped structures. So again, there's another one over here, and so on and so forth as they continue out. We'll focus on just one. These liver lobules are made up of rows of hepatocytes. So basically, these hepatocytes, the cells, oops, a little too far. So we have these rows of hepatocytes that line up, radiating out of the center of this lobule. So I have some going that way and some going this way. And I'm just going to draw the columns now. I'm not going to bother drawing the dividers because it's going to take me too long. I'll put 2 here just for the heck of it. Not just for the heck of it. You'll see why in a second. And I'll put some dividers in that one. So these rows of cuboidal hepatocytes line up, radiating around the central region of this lobule. And in the central region of this lobule is a structure known as the central vein. Remember, we know that the liver is one of the major jobs of the liver is to be able, and actually let's make that smaller so it fits better, is to process all of the blood that is coming from our digestive system. So we have that blood coming from the digestive system via the hepatic portal vein that is carrying blood that is very nutrient rich. Of course, to get that nutrient in the small intestine, as the small intestine has absorbed this, as we're going to see, that is primarily an active process requiring ATP. So while this blood is nutrient rich, it is also going to be oxygen poor because most of its oxygen will have been used up. So let's go ahead and show you a branch of this. These branches of these hepatic portal veins are going to be located out here, in the connective tissue septums of our lobules to feed this blood into highly specialized, And we'll use purple for this. Big, loose, incomplete epithelial capillaries that are our sinusoid capillaries. You remember way back in the cardiovascular system, we talked about how sinusoid capillaries are big, huge openings with incomplete epitheliums, which allow them to move large things into and out of our blood supply. Now remember, this is where food is coming back to our body from our digestive system, right? And maybe you picked up that cheeseburger that dropped on the floor because you have in your house have a 5-second rule. But does that mean that no bacteria got in it during 5 seconds? No. So we do have a concern that way. Luckily for us, inside these sinusoid capillaries are anchored to the walls of the capillary on the side are going to be some special cells that are known as Kupffer cells. These Kupffer cells are phagolytic cells that are capable of finding pathogens, grabbing them, and then presenting their antigens on their surface so they can act as antigen presenting cells. So there should be a P in there. There we go. And they can help to store, for instance, heavy metals and lipid-soluble vitamins and things along those lines as well. So they do provide some storage as well, and they're located in those sinusoid capillaries running between the hepatocytes. So the hepatocytes can do their job of making the blood nutrient appropriate. They can store extra glucose. If there's not enough magnesium, they can release magnesium into the blood. So notice when the blood reaches that central vein,
Did you say the hepatocytes make the blood nutrient appropriate?
yep. So one of their major jobs is to store excess nutrients and release nutrients that are deficient. So we go from blood that was nutrient rich to blood that was nutrient appropriate. Again, I know this is hard to understand because I know all of you work really hard to have a completely balanced breakfast that has all the right amount of proteins and micronutrients and vitamins and minerals and all those things. But some people eat a donut and Diet Coke for breakfast. And they're not necessarily getting all of their, maybe rich in nutrients like glucose, but it's going to be deficient in others. So again, remember we talked about how there's this magic that happens that has to occur in the liver. This is where the magic is happening. The magic is happening with those hepatocytes being able to absorb nutrients or release nutrients to make the blood nutrient appropriate. However, this requires a lot of work, and work typically requires energy. And notice that pesky hepatic portal vein ain't got no oxygen in it. If only there was a systemic blood vessel, right? Because we know those arteries contain a large amount of oxygen in them that could possibly bring oxygen to the party here to help us to do work. Does something like that exist? Did we learn about a systemic artery that maybe comes off of the celiac trunk and provides? Yeah, exactly. Right, we had that hepatic artery that comes off of the celiac trunk, and it contains oxygen-rich blood, bringing that oxygen to the party. And this hepatic artery happens to also be located here in the junctions of our septum between the liver lobules. So notice two blood supplies come to the liver. We have nutrient-rich blood coming from the hepatic portal vein. We have oxygen-rich blood coming from the hepatic artery, and they are both feeding into these sinusoid capillaries, where that blood is then mixed and processed, becomes nutrient appropriate as it enters this central vein. And this central vein feeds into the hepatic vein. And where does the hepatic vein go?
Vena cava.
There you go. So we're going to talk about the functions of the liver a little bit later, but here is the anatomy that allows it to do this work. Oxygen-rich blood is brought by the hepatic arteries. Nutrient-rich blood is brought by the hepatic portal veins. They mix together in the sinusoid capillaries. Our hepatocytes move nutrients back and forth between the blood and storing them themselves, making the blood nutrient appropriate, depositing in the central vein, which sends it into the inferior vena cava, which of course sends it into the right atrium. And once that nutrient-rich blood gets the right atrium, it goes to the lungs to get oxygen, and then out on its merry way to the body. But these hepatocytes are not underachievers. Definitely filtering the blood is a full-time job. However, they like to be well-rounded. They like to have hobbies as well. And one of their favorite hobbies is to make bile. So let's not forget that these hepatocytes need to be making bile. They're constantly making bile. When they make this bile, whoops, hold on. Shucks, I didn't want that to be green, but that's all right. When they make that bile, they feed that bile into these tiny little canal-like vessels, bile vessels, called bile caliculi. These bile caliculi feed the bile into bile ducts. And these bile ducts are also located here in the connective tissue septum of our liver lobules. So notice in this case, we are making that bile and feeding the bile outward in the opposite direction of the blood, out of the hepatocytes to those bile ducts. And so notice at the corners of our liver lobule are these three important structures known as the triad. And this triad includes three structures, right? A branch of the hepatic portal vein, a branch of the hepatic artery, and a bile duct. And these three structures collectively form our triad, which, if you notice when we go back to our illustration, we'll get to that in a second. Excellent. Notice here we have this beautiful 3D illustration that does a nice job of showing this for us. Again, here at the corners, we have that portal triad including a branch of the hepatic artery, a branch of the hepatic portal vein, and a bile duct. Notice the artery feeds oxygen-rich blood into the sinusoid capillaries. The portal vein feeds nutrient-rich blood into the sinusoid capillaries. And notice the sinusoid capillaries are just surrounded by rows and rows and rows of hepatocytes. Notice here, anchored to the wall of our sinusoid capillary, we have one of those Kupffer cells we talked about. And oxygen-rich blood mixed with nutrient-rich blood is processed by the hepatocytes and fed into the central vein. The central veins come together and feed into the hepatic vein and out of the liver. At the same time, the hepatocytes are making bile, feeding it into the bile canaliculi, which feed it out into the bile ducts to leave the liver and either go into the stomach, into the small intestine, or to be stored in the gallbladder. Here we see it with a nice illustration, another nice illustration, again, showing those portal triads in the connective tissue septums. We did all that. We'll come back and do all this again, but I want to look at the pretty pictures first. Here, we see it histologically. Notice we can relatively clearly see the septums that are dividing. Notice this one looks a little bit more square than it does, but again, it's about how it was put on the plate. But we can clearly see the central vein at the center, the spaces that are the sinusoid capillaries between the radiating hepatocytes, and most importantly of all, because I guarantee you're going to be responsible for it on the exam, we can see the portal triad. And if we take an even closer look, central vein, hepatocytes, sinusoid capillaries, right here we see the portal area, but let's take a closer look at that portal area. Not only do we know the three things that are going to make up the portal triad, but most importantly of all, we can identify them histologically. And conveniently enough for you here, I've labeled them 1, 2, and 3. Which of our three portal triad structures do you think one is?
Vein.
Right. How did you know?
It's bigger because it holds the nutrients.
Not a bad guess. It is bigger, but remember, when we talk about arteries and veins, veins typically have a lower pressure in them than arteries do. So as a result of that, they need a larger lumen so that it's easier for them to move their materials. So notice of the three structures, it absolutely has the largest lumen. Notice it also, we can see the simple squamous epithelial tissue that is forming its endothelium. But do we really see the muscularis of this, the tunica, the tunica muscularis, the smooth muscle? Not so much so. Compare it to 2. Notice 2 also has a simple squamous epithelial tissue lining it, but this one, we can much more clearly see the smooth muscle of the wall. And it has a much smaller lumen to it. And conveniently enough for our purposes, it actually has red blood cells centered in the center of it. So what do you think 2 is?
The artery, portal artery.
Yeah. Again, we don't want to call it the portal artery. It's the hepatic artery. Right? It's the portal vein, but it's just the hepatic artery. Absolutely. Of course, by default, that we know what 3 is, but also notice the big difference about 3. Where 1 and 2 is lined by a simple squamous, being a duct, notice it is instead lined by cuboidal cells. And when we see that structure lined with cuboidal cells, that clearly is a duct. And so 3 is indeed our bile duct. So what's this one over here?
Bile duct.
Duct.
Not a bad guess, but muscular wall, Squamous cells, notice squamous cells so thin that the nuclei stick out, right? They're not cubes like this. So this is indeed, and here I'll erase it so you get a better view of it. This is actually a small artery. Again, here the duct is very, very clear. All right. I think, do I have one more? Nope. Notice here at a lower magnification, it can be a little bit trickier to tell most things, but notice it's still pretty easy to tell. Well, let's not use blue. The veins. Hepatic portal veins are typically the largest structure. So even at this low magnification, it's pretty easy to tell where the hepatic portal veins are. This is likely the artery. This looks a little bit like the duct there, but again, at this at this lower magnification, it can be trickier to tell the structures. It's much easier at a closer, higher magnification view. But like I said, even at this low magnification, it's pretty easy to tell the veins because they have such large, prominent lumens to them. All right, so absolutely positively you are responsible for those things histologically, but they should be pretty easy to tell and distinguish because of their distinct characteristics. All right, let's go back all to pretty words. We did all of this on my illustration, but I wanted to go through it again. Remember, the hepatocytes are, again, total worker bees. Their job is to make the blood nutrient appropriate. They also are going to detoxify the blood, get rid of toxins in the food you eat, get rid of medications in the blood, get rid of antibodies, get rid of hormones, right? Detoxifying all of those chemicals and taking them out of the blood, which is one of those reasons I've never understood people who eat livers. Livers are the garbage can of your body where that stores all the junk that you want. So that doesn't make me want to slice it thin and fry it and put onions on top of it. I don't get people who eat livers. Again, if you eat liver, more power to you. But I don't quite understand it. No, even that's gross. So it does all that work. And like I said, because that's not enough, it also produces our bile. It's got that hobby on the side. Again, as we mentioned, the sinusoid capillaries contain those Kupffer cells. They're phagocytes that are antigen-presenting cells to activate our immune response. But remember, they also will help to store some heavy metals and some lipid-soluble minerals that are necessary in our body. So there you go, iron, lipids, heavy metals, stuff like that. We looked at this picture, and again, we looked at it here, that nice hexagonal shape with that connective tissue septum in between, central vein at the center, hepatocytes radiating out, separated by the sinusoid capillaries, bunch of Kupffer cells that they show us in this one here. And again, at the junctions, we have that portal triad, the hepatic portal vein, a branch of the hepatic artery, and our bile duct. We've seen the anatomy. We also talked about the function. Two blood supplies coming to our lower lobule. Hepatic artery that contains what again? What's the hepatic artery bringing to the party?
Oxygen rich.
Oxygen.
Oxygen, absolutely. And what's our hepatic portal vein bringing?
Nutrients.
Nutrients, excellent. So those are what they're bringing to the party. They mix in that sinusoid capillary, and then again, become nutrient appropriate, leave out the central vein to the hepatic vein, oxygen poor, nutrient appropriate, and on its way back to the heart. Again, movement between the hepatocytes and the blood is indeed two ways. If it is deficient in a resource, it can release it. Right. So if you're not getting enough magnesium in your blood, your liver stores, in your diet, your liver stores magnesium and can release it. But what happens if you don't have enough magnesium in your diet tomorrow and the day after and the day after that? Right.
It eventually depletes.
Yeah, eventually you're going to deplete the stored resources, at which point you then become malnourished. right? Because you're no longer able to, you no longer have the stored reserves of those micronutrients that are necessary. And so as a result of it, you become deficient in those resources. So it is important to have that well-balanced meals with all of the, vitamins and minerals and resources that we're not capable of producing on our own. And like I said, because the hepatocytes are totally type A, they also make our bile as well. And again, blood flows inward, bile flows outward. We have those opposite flowing directions of their material. And we did all of the histology of that. The last thing I want to do for today is talk about what happens to that bile once it is produced. Because we have actually, that's not true. I have two things I want to do. What's after this? Perfect. Excellent. All right. Anatomy. Notice coming out of the liver, coming out of the right lobe of the liver and the left lobe of the liver are the right and left hepatic ducts. And their name tells you everything, right and left, because they come from the right and left lobes of the liver. Hepatic duct. Notice they feed into the common hepatic duct. Common because it contains bile from both the right and the left side, but it's still hepatic. Now, as we mentioned, if we want to release this directly into the small intestine, we are capable of doing that. But if you're not needing it, then what we can do is that bile can move up the cystic duct to be stored in the gallbladder. And as we talked about, not only will it be stored, but it will also be concentrated here in, I don't know why that's so small, here in our gallbladder until it's needed. With the help of cholecystokinin, our gallbladder can constrict, and when it constricts, that bile will then come out of the cystic duct. Notice the cystic duct has the ability to transport bile in two directions. It can go into the gallbladder, it can go out to the gallbladder. It is the only duct in this process that can move bile two ways. Notice for all of the hepatic ducts, it's all coming out. And notice that bile coming from the gallbladder and bile coming from the liver all mixed together into a single duct because it contains bile from both the liver and the gallbladder is known as the bile duct or the common bile duct. And it is that bile duct that, as we talked about, feeds into the hepatopancreatic ampulla through the hepatopancreatic sphincter, out the duodenal papillae into the small intestine. So notice 1, 2, 3, 4, 5 different ducts named precisely by their function. Right hepatic duct comes from the right lobe of the liver. Left hepatic duct comes from the left lobe of the liver. Common hepatic duct from all of the liver. Bile duct contains all bile. And the cystic duct is the special one, the two-way duct that can carry bile to the gallbladder and also out of the gallbladder. All right, questions on that?
So what is the means by which it goes either up?
So great question. Going up is primarily based on a fluid gradient. If you think about it, if this sphincter is closed, bile is going to back up. And as bile backs up, that pressure gradient, because new bile is constantly being produced. So we have pressure of bile coming this way. We have the backup of bile coming this way. And so that pressure gradient basically forces it back into the gallbladder. However, remember our hormone cholecystokinin does two things. It opens the hepatopancreatic sphincter and it causes a contraction of the gallbladder. If you or anybody you've ever known had gallstones, when it is most painful is when you eat a very lipid-rich meal. You have that 3-scoop ice cream sundae, and suddenly you're getting that pain in your side. And that pain you're getting in the side is because as the gallbladder is constricting around those gallstones, it's painful. So what causes gallstones? Gallstones are caused by a solidifying of the bile, a crystallization of the bile to form bile crystals. Why it happens is not fully understood. We know there's a genetic predisposition, so it tends to run in families. If your grandmother had gallstones, it's more likely that you will have them. But it's also very related to diet. A very lipid-rich diet can lead to an overproduction of bile, which can cause more to be stored and can cause it to crystallize. But also a zero lipid diet can also cause the accumulation and lack of release of bile from your gallbladder, which can lead to the ultimate crystallization of it. So both a lipid-rich and a lipid-deficient diet can potentially cause gallstones to form.
If you don't have a gallbladder, where does the bile go? If it can't be stored and concentrated, where does it go?
So remember, as we talked about, what will happen is that the ducts of the liver will enlarge, allowing it to be able to store some additional bile more than it normally would inside of it. So it will store more there, but remember, it's not going to be able to concentrate it the same way that the gallbladder is. So while you can store it to have some to release, it's not going to be as efficient. All right. All right, this is the anatomy. And again, I want to talk in here again, we see that common bile duct feeding into the duodenal ampulla, duodenal papillae, controlled by the hepatopancreatic sphincter. I want to talk about liver functions, but I think I want to save that for next time. What I want to do instead, because again, I think there's some challenges involved with this. And if you've looked closely, as I'm sure you have at your Canvas site, and if you haven't, first and foremost, shame on you. But if you look closely at this, you'll see that there are some pictures of some models. These are models that I usually pull off of the wall when we're talking about this right now, because I guarantee they're going to be on the exam. Of course, we unfortunately don't have the ability to look at those right now. So instead, we conveniently enough have pictures of them. So let's look together at that. Under your modules, under the lab handouts, you'll see a couple things. The first thing that you'll see here, obviously, is your study guide that is going to provide all that information here. But here is this great model of the liver. Now, this is kind of a weird posterior and inferior view. They've kind of cheated a little bit here. Notice here is our inferior vena cava. So that we know should be posterior. But they've cheated a little bit with our lobes. So notice here, this view, they've allowed us to see all 4 of our lobes. We have over here, the much smaller left lobe. Over here is the right lobe. Then here we have our caudate lobe. And this cross-section piece right here is the caudate lobe. Notice also, since we just finished talking about them, we can also very, very clearly see, what color is going to show up the best on this? Let's try pink. Notice this would be the right hepatic, the left hepatic duct coming from the left lobe, the right hepatic duct, which feed into the common hepatic duct, which can either feed into the cystic duct, which feeds into the gallbladder, or the cystic duct and the common hepatic duct can feed into the singular bile duct that is going to carry it out. So notice all 5 of our ducts are located here on this model really, really nicely. All right. So we have our bile-related ducts, and of course we have some branches of the bile ducts. We don't see the canaliculi. We're not going to see that at this magnification, but we can see the bile ducts collecting it from all the lobules all throughout. So questions on those? Those are the easy ones. Pretty simple, straightforward. Any question on that before we go forward? Oh, cool. Look, notice also they've given us our ligamentum teres coming off the bottom there. And if you flipped it around to the front, you'd see the falciform ligament as well. All right, so those are the easy structures. Questions on that? All right, notice the other thing we need are blood vessels. But the weird thing I notice about this is I actually see 3 different colored blood vessels. I see red blood vessels like I expect to see. I see blue blood vessels like I expect to see. But there's also this weird white blood vessel. Why do I have 3 different covered blood vessels going on here?
Rich, poor, and mixed.
Close. You've absolutely got the right idea. Let's start easy. What do you think blood vessel 13 is right here?
Artery with oxygen rich.
So which one would that be? Which artery?
Hepatic.
Yeah, hepatic artery. I don't know if you said hepatic artery, but you might not have gotten it out when you hit your mute. I only heard artery. Absolutely. So that is that oxygen-rich hepatic artery. And let's make this smaller so I have room for all of this. Excellent. Let's start easy as well. What do you think 11 is?
Portal.
What do you think 11 is?
Hepatic vein.
Not a bad guess. 11, though, is that big, huge one that's not
Inferior
into
vena
the
cava?
liver. Yeah, it's the inferior vena cava, which we know is nutrient appropriate and oxygen poor because it's on its way back to the heart. And along those same lines, as you guys notice 23 that feeds into the inferior vena cava. That would be the hepatic vein. So 23, I assume you guys can see the 23, but 23 right here, this right here, the blue, is that hepatic vein, again, coming out of the central vein, carrying the blood back. So then what does that make 12?
It's coming from the intestines.
Exactly. And what was the blood vessel coming from the intestines? That is going to be nutrient rich, but oxygen poor.
Portal vein.
Exactly. It is the hepatic portal vein. So notice what they've tried to do here is show us the different qualities of the blood vessels. 12 represents that hepatic portal vein. Notice it's bringing its blood to the lobules, right? The hepatic artery is bringing its blood to the lobules so that we're bringing the oxygen, we're bringing the nutrients to be processed. And then once processed, our hepatic veins carry it out to the inferior vena cava and carry it out. So that's why they've given us this kind of, and it's actually, when you see it in person, it is more of a light blue than a white. And so that light blue is trying to remind you, hey, this is a special vein. It is a vein, but it's that special hepatic portal vein, bringing that nutrient-rich blood that needs to be processed. So 2 blood supplies in, right? Hepatic artery and hepatic portal vein, 1 blood vessel out, hepatic vein into the inferior vena cava, right? Liver's like Thunderdome. All right. Questions on that? Excellent. Let me show you one more thing. My favorite model, which of course should be a dead giveaway. You're going to be responsible for this on the exam. Here's the key to it. Here is the picture. Awesome. Oh, it doesn't even fit on the screen. That kind of stinks. All right, we'll start at this end. Whoops. Stop it. There. What they've done with this picture, and here I'll cheat. Go back to this real quick. What they have done with this picture is basically they have taken a liver lobule like this, and they've taken a wedge of it, just like that. So notice when we look at this picture, we see our portal triad, including 1, 2, 3, 4 structures. Wait, what? All right, let's see if we can figure this out. Structure 1 in red, what do you think that is?
Hepatic artery.
There you go. Structure 2, big blue thing.
Portal vein.
Hepatic portal vein, excellent. Structure 3, green thing.
Duct or
Bile
bile.
duct. Yeah, absolutely. So then what the heck's this 4th thing? An idea?
Capillary.
Not a bad guess. Nope, it's a nerve. So it just represents a nerve going through there. I don't know why they put it there. It's not a part of the portal triad. It's not called a quadrad. It's called a portal triad. So feel free to ignore it. But again, artistic license, our artists put it in there. But notice, as we expect, our oxygen-rich blood from the hepatic artery, the nutrient-rich blood from our hepatic portal vein are fed into the sinusoid capillaries. Notice they've kind of tried to draw the incomplete epithelial cells of this to show us that this is a sinusoid capillary as it's going through there, going between the rows of hepatocytes. Notice also the hepatocytes have their bile canaliculi that are collecting the bile and bringing that bile out to the bile duct. So remember, bile moves towards the edge, whereas our blood moves inward. And notice if we follow our blood all the way to the middle, then lo and behold, in the middle, we have our central vein that collects the now nutrient appropriate oxygen poor blood and ships it to the inferior vena cava. So all they've done is taken us a nice little wedge of that liver lobule to show us it from the outer surface where the triad is towards the center where the central vein is. So it's a really, really nice. Can I decrease? I don't know if I can decrease the magnification of this, but that's all right. You can see it on your own. Great model, does a great job of showing all these things, and I guarantee you need to know this because this will be on the lab exam. All right, so both of these models are things that I've provided for you, which again should be a strong indication to you that you are going to be responsible for them on the lab exam. All right, questions on that? Of course, you also have your handout of what you're responsible for on there, so that is good as well. All right, questions on that? How are we on time? We're doing good. Okay, excellent. Here's one last thing that I want to talk about. So let's cheat. Hold on. So in the next class, we are going to talk about the functions of the liver, functions of bile, all of that kind of stuff. But here's the last thing I want to leave you with to think about for today and over the weekend. The last thing we have to do in the small intestine is talk about our absorptive pathways. Your goal in life is to eat a cheeseburger. And once you eat that cheeseburger, the goal is to get the nutrients of those cheeseburger to your heart. So once you get it to your heart, it can be distributed everywhere throughout your body. Of course, that includes water, and most importantly, carbohydrates. Oops, wrong button. Carbohydrates, proteins, and lipids. We are going to describe these three pathways. And I guarantee one of these three pathways is going to be an essay question on your exam. All right, I already know there is a test bank of 3 questions, one for carbohydrates, one for proteins, and one for fats, that I guarantee you will be getting one of those 3 questions on the exam. And you're going to be responsible for describing all of the ways that macromolecule is broken down mechanically, all of the ways it is broken down chemically, all of the specific enzymes involved in that process and where they're produced. We're going to talk about how it crosses that absorptive cell, both on the apical surface and the basal surface, what it goes into, whether it goes into a blood vessel or whether it goes into a lymphatic vessel. And luckily, because we've had the cardiovascular system, and luckily, because we've had the lymphatic system, we will also be able to trace the pathway of that nutrient all the way back to the right atrium of the heart. So we will do that all together on Monday, but Monday is also 2 days before the exam. So I would strongly encourage you not to wait till then to look at this for the first time. I will remind you, this is your big study weekend. You have the textbook, but most importantly, you have my outlines. My outlines tell you exactly what we're going to be talking about on Monday. So there is no reason over this 5-day weekend that you should not look at those things and start preparing yourself for the things we're going to talk about on Monday. Because if all of this stuff is fresh and brand new to you on Monday, that's not giving you a lot of time to process and synthesize and make sense of it. So I strongly, strongly, strongly encourage you to look at the lecture outlines, see what we're going to be talking about in this last section, and prepare appropriately. All right? Questions on that? All right, excellent. That is everything that I wanted to cover for today. And we finished 10 minutes early. Look at that, bonus. Excellent. All right, you guys have a very good, very safe weekend. And I will see you guys on Monday. Have a happy and safe St. Patrick's Day.